Good morning. Jane and I would like to welcome you to the second annual Cellular and Molecular Biophysics Conference. In coming here from Berkeley, I came to realize that Houston is an Athens for biophysics. And today we have both some uniquely strong speakers, but also by having all of you join us, I think a real chance to have strong interactions and new collaborations and new discussions that span from molecules to cells. So please be sure to join the discussions and also to take advantage of the Barella features, which Dawn is going to explain to you. So go ahead, Dawn.
Good morning, everyone. I'm going to share my screen real quick. Okay, this is the Barella homepage. If you click on the People tab, you can choose the categories of people that you'd like to network with, people who have similar interests to you. You can also suggest meetings. If someone accepts a meeting, you'll be able to chat, have a video chat with them face-to-face through your computer anytime during the conference. And the last thing I want to show you is, as you can see, the videos are going to be in the stream tab here. Please feel free to enter any questions you have for the speakers in the chat on the right side of the box. And I think that's all I have for now.
So I guess we should get started with the first session on single molecule methods. Michael?
Okay, welcome. For the first section, I guess I'll start as a practice with Barella, and this is a new sort of a style for all of us. The questions, if you have any question, please enter them in Barella. And we do encourage the trainees to ask questions, but in order to know that you are a trainee, graduate student, or postdoc, I guess you can put a note next to your question, and this will prioritize your questions. So at the end of the talk. So the first talk is going to be given by Dr. Ilya Finkelstein, who's from University of Texas in Austin, and he's an expert on single molecular studies of everything related to DNA transcription regulation. That includes CRISPR-Cas9. So Ilya, you have the floor.
Thank you very much. Audio check, you all hear me? Nod.
Yes, we hear you.
Wonderful, thank you. Yeah. So before I get started, I want to thank the organizers for putting this together. Looking at the amazing lineup of speakers from Houston and beyond, I realized that Austin is really almost like the, I guess, the Marfa of biophysics. We have a few people, but we really need to build a really big center like Houston, and we'll work on that. So let's see. Today I'm going to tell you about the CRISPR side of my lab, but I also want to mention that my lab studies DNA repair, specifically focusing on mismatch repair and homologous recombinations. Homologous recombination, we use a bunch of biophysical tools, including single molecule methods, which you'll be hearing about today. But today, I'll talk about the second love of my lab, which is CRISPR-Cas biology. And when I got into this field, I quickly realized that we'll all soon be CRISPR-Cas biologists. If you just look at PubMed, and the number of PubMed publications is growing exponentially. This is a semi-log plot shown in yellow. And the number of publications with the keyword CRISPR is actually growing also exponentially, but much, much faster. Sometime by 2027 or so, we'll all be studying CRISPR-Cas biology. So I think it's good to get started now because you have a few years to catch up before all of us have to do it. Anyways, so I saw that light a little bit early when I started my independent lab at UT Austin, and we got into CRISPR-Cas biology. I'm just going to remind you all about what CRISPR-Cas is. It's an adaptive bacterial and archaeal adaptive immune system. Here's the three main pathways that comprise the CRISPR-Cas adaptive immunity. Many of us are familiar with Cas9. Cas9 is an example, not even the most common example, of the interference side of CRISPR-Cas adaptive immunity. And during interference, a foreign DNA, for example, here illustrated as a phage, injects its genome into the host cell, into its target cell. And then a surveillance complex uses a CRISPR RNA that's complementary to the target DNA or RNA, finds this CRISPR RNA, or this target DNA, this invading pathogen DNA, and then marks it for degradation. Here, I'm going to be talking about type 1 CRISPR-Cas complexes, specifically the type 1e complex. Type 1 CRISPR-Cas systems are the most common type of systems in bacteria and archaea. So the type 2 Cas9-like systems we've all heard about are actually pretty rare. Cascade is the surveillance complex. It's about half a megadalton of multi-protein complex that I'll show the structure of later. But this is the surveillance complex that identifies the foreign DNA for destruction. It does not actually encode any enzymatic activities of its own. It's not an ATPase. It recruits a helicase nuclease called Cas3. And then this helicase nuclease Cas3 is the module that actually degrades the foreign DNA. But of course, adaptive immunity has to be adaptive. So what does that mean? There's additional modules that can acquire foreign DNA into the CRISPR locus over here, where that information is stored as genetic memory for targeting new phages. The Cas1 and Cas2 integrase proteins are necessary and sufficient for this adaptation. It's called naive acquisition. These are related to transposon-like proteins, and they can take short 20 to 30 nucleotide pieces of DNA from the virus and put it into the CRISPR locus near the leader here sequence. Now, naive acquisition is very, very inefficient. However, genetic and cell biological experiments have shown that, in fact, there is a much more efficient way to acquire memory, which is called primed acquisition. And this memory is really active when a related phage, a phage that is related to what's already in the CRISPR locus, tries to invade the cell. So for example, if the phage is mutating to avoid interference, makes a few point mutations to not be complementary to the CRISPR RNA, primed acquisition can still acquire that phage sequence very rapidly. And that primed acquisition genetically requires the Cascade surveillance complex and Cas3. Okay, so this is kind of an interesting conundrum. I'll talk to you about what our views on this particular very large megadalton scale assembly in CRISPR-Cas adaptive immunity means. So this is a single molecule session, and I've been a single molecule biophysicist for quite some time, so I wouldn't be complete without bashing single molecule biophysics. And so there's been a lot of biophysical tools to probe CRISPR-Cas adaptive immunity, including the type 1 complexes. There's been some pioneering work using magnetic tweezers, where people like Ralph Seidel's lab and others have looked at how the Cascade surveillance complex identifies a protospacer adjacent motif, opens this R loop, and this R loop then propagates. This was a key finding. It propagates from the PAM directionally away from the PAM. And it's dependent on negative supercoiling and so on and so on. And then Cas3 is recruited to this open R loop complex. Single molecule fluorescence has been useful to address this kind of question as well, including how this R loop is open, what the rate of R loop propagation is, and what the role of mismatches in R loop propagation in the target DNA relative to the CRISPR RNA mean for R loop propagation. And there's a flavor of single molecule imaging, which we do, called DNA curtains, which has also been used to address some of these same questions. Okay, so all of these tools have provided very important insights into how Cascade recognizes the protospacer adjacent motif via the CSC1 subunit, how that causes opening of the DNA, and how that DNA pairs with the CRISPR RNA. But there's one major disadvantage of all of these single molecule tools. They all probe a limited nucleic acid sequence space. What I mean is my postdocs would probably mutiny, or graduate students, if I asked them to do this experiment on 10 different DNA targets. Maybe with single molecule, with a very patient experimenter, you can get through 10, but you can't get through 100. Right? So I've always worried that these tools are very high resolution, very useful, but the trees may be obscuring the view of the forest. Can we look at this globally? So I'm going to give you a little diversion before I get to the single molecule part of my talk, but I'll give you a little diversion about how we're trying to merge single molecule biophysics with modern high throughput approaches, including sequencing. Okay, so the next few slides are really not single molecule, although I do believe we can push us into the realm of single molecule as well. So what did we decide to do? We adapted work from others and recognized that next generation sequencing via the Illumina platform, Illumina of course controls something like 85 to 90% of the DNA sequencing market. This next generation DNA sequencing produces two outputs. It produces a file, which is what every user cares about. This file has the sequence of every DNA molecule that they wanted to sequence, ideally. And then the second output is a microfluidic chip, which has very nice imaging properties designed for fluorescent imaging. And that chip has spatially registered DNA on it. What I mean by spatially registered is there's a bunch of pieces of DNA on this chip, and the XY coordinates of all of these DNA molecules are recorded in that sequencing file, but that column is always thrown away by users because they don't really care whether DNA is on this chip. Now, these chips are basically thrown into the trash. So what we did was we upcycled these chips for biophysical studies. So a brief primer on how Illumina-based next-gen sequencing works. Okay. So first, our DNA library is paired with oligonucleotides that are called adapters. These adapters can then be, the DNA is denatured and grafted onto the surface of a flow cell. The flow cell also has oligos on it that are complementary to these adapters. Then through several cycles of colony PCR, so this is essentially bridged DNA molecule between two priming sites, and then PCR happens many times, and you have amplification of a single input DNA into a cluster, which is called a colony for a PCR colony. So these are all the same DNA molecule, just amplified to about 100 or so same DNA sequence that are amplified to about 100 different molecules, not single molecule. And then this appears as a nice fluorescent spot on the surface of the flow cell. And then of course, in Illumina sequencing, each of these fluorescent spots is imaged with different color DNTPs during synthesis by a polymerase. So, you know, one color goes red, one color goes yellow, and so on. And then by doing step-by-step synthesis, you can decode the sequence of each of these DNA molecules. Okay. So what we did was we allowed Illumina to do its thing, create these chips, sequence the DNA, and then we took these DNA molecules and then mounted them into our single molecule fluorescence microscopes to see what we see. Okay, and that's where we start. So our experimental strategy looked something like this. Go in the trash bin and dig up these different chips and then stick them on our microscope slide. I actually had a hard time convincing anyone to do this because who wants to start their project looking through garbage? But luckily, I had a visiting scientist who didn't know any better. He was trying to get into biology. He was a mechanical engineer, good at imaging. He didn't know any better, so he just did it. And eventually he convinced a postdoc to join him, and together they were a formidable team. So when we took these chips out of the garbage, they looked like garbage. So here on the bottom is a fluorescent image in multiple channels of these chips straight out of the garbage. And you can see there's a lot of little dots. That's promising. But all of these dots glow in purple and green and so on because there's all this fluorescent gunk left on the surface. We cleaned up the fluorescent gunk by chemical treatment, denatured the DNA, removed the non-template strand, and the template strand remains covalently attached to the surface. Okay. We added fiducial markers by hybridizing with fluorescent primers in some positions. We can talk in detail about what that means. And then we resynthesized through one round of synthesis. And then we could add our protein of interest here, this fluorescent cascade molecule that targets some of these DNA clusters on the surface of the cell, on the flow cell. The cascades are labeled with a different fluorescent color from the fiducial marker. And then we wrote an image analysis and bioinformatics pipeline that could then take all of these fluorescent images and using the sequencing output from Illumina, decode the exact DNA sequence at each position on our flow cell. So now we know what each of these dots means in terms of DNA sequence. Okay, so we call this CHAMP for Chip Hybridized Affinity Mapping Platform. This was first published in 2017. We just had another paper extending it further out in Nature Biotech, I think last year, in the pre-pandemic times, I think. And we have another paper coming out soon, I hope, that will extend this to RNA binding proteins as well. Okay, so what does the data really look like? So for example, here we are just doing a titration of this cascade surveillance complex. As a concentration of cascade increases, the brightness of these clusters also increases, but some of them get brighter than others. For example, the ones in red get brighter and brighter. The ones in blue here highlighted don't get brighter. And these are different sequences. And I'm highlighting the blues are all the same sequence, the reds are all the same sequence. And you can see that they have different brightness, meaning there's different affinity. So from this, we can calculate, essentially, this is a very, very high throughput EMSA, a gel shift experiment. We can also measure on rates, off rates, and protein-protein interactions, as I'll show you soon. So using this kind of approach, we can start mapping what is the sequence preference or the mismatch tolerance for some of these cascade surveillance complexes, and then in subsequent papers, other CRISPR-Cas systems. I'm going to summarize a lot of data very quickly because I want to get to the single molecule side of the lab. But here is just one example. There's a lot of data. There's tens of thousands of different DNA sequences that we probed on one chip. But here's an example of a typical library that we like to use. In this library, we've randomized 6 nucleotides that comprise the protospacer adjacent motif, or the PAM, that's recognized by the CSE1 subunit. I'm just showing the last 3 here. And we've also created what's called a quote unquote doped target library. So this is what's complementary to the CRISPR RNA part of Cascade. And then we've basically injected about 10% randomness at each of these positions. So we can measure every single mismatch in the target library. We can measure every single pair of mismatches in the target library. And we can measure some combinations of 3 mismatches. In addition, there's other sequences that we've included as controls, like scrambled and so on. And so here I'm plotting essentially the log of the binding affinity for each of these sequences relative to the reference sequence shown on the bottom. The PAM is the first 3 nucleotides, and then the CRISPR RNA complement is shown to the right of that. And you can see what a single substitution in any of these positions does. Okay, so the higher the bars, the higher the binding penalty, the lower the overall affinity. Okay. And this is all a reference to the perfectly matched sequence. So 0 would be perfectly matched. All right, so what you can immediately see is that at every 6th position, there's essentially no penalty for having any kind of mismatch. That's what you can see from this kind of quote unquote noisy data. This isn't noise, this is real data, but you can see the noise as the error bars here. But this is what you see, and you can even see some positions having a mismatch may stabilize the complex on the DNA. There's lots of data. We need to reduce dimensionality, so we build models. This is a very simple model that essentially computes a mismatch penalty that's dependent on the type of mismatch, for example, a T to a C or a T to a G or a T to an A kind of mismatch, and multiplies it by a weight. So this is a linear additive model. It works really well. We've done it for multiple targets. Also allows us to combine different DNA sequences so we can look at them in aggregate. And essentially you get an output that looks like this. This is the penalty of having a mismatch at a certain position. And this is the position. The PAM is again shown in the first 3, also coded pink. And then you can see this penalty for mismatches that proceeds across the CRISPR RNA. And we see a lot of features that were already known from prior single molecule and structural experiments. So first and foremost, we see the 6 nucleotide periodicity, where having a mismatch essentially produces no penalty at all for cascade binding to its target DNA. That's curious. We'll tell you why that happens. We also see, as expected, an overall loss of sensitivity to mismatches as you proceed farther away from the PAM. And this is consistent with this directional R loop propagation away from the PAM. Once you form 8 or so nucleotides of RNA DNA base pairing, essentially the rest of it doesn't matter so much. The complex is quite stable. And finally, we see what was not seen before, or what was seen but ignored before, was a very clear periodicity that's also a 3 nucleotide periodicity, where the penalty doesn't go to completely 0, but does decrease substantially. Okay. We see a bunch of other interesting things, but I just want to give you a flavor for how we interpret data like this. So here we come back to our structural biology colleagues and look at the structure. This is the structure of Cascade. Cascade looks like a seahorse. That's how it was labeled or named by the people who saw it first, using the low resolution EM. So this is a seahorse structure. Just walking you through it, the CRISPR RNA is in purple. It's occluded by the protein around it. in blue, the head of the seahorse is the CSC1 complex. I'm sorry, it's CSC1 protein. CSC1 is critical. We'll talk about it shortly. It binds and recognizes the PAM. It opens the DNA adjacent to the PAM for R loop propagation. And it also interacts with Cas3 to recruit that helicase nuclease for targeting the DNA for degradation. Then moving away from CSC1, there's 6 repeats of the Cas7 subunit that templates itself across the CRISPR RNA, 1, 2, 3, 4, 5, 6 of these subunits. What you're not seeing is the Cas5 on the back end of the complex, the CSC2 also on that side of the complex, and then the capping Cas6 that caps a hairpin on the CRISPR RNA distal from the CSC1. I want to draw your attention to the Cas7. This 6 nucleotide and 3 nucleotide periodicity really comes in from the Cas7, the 6 Cas7 protomers. Okay, so the 6 nucleotide periodicity was known before from structural studies. There's a finger that Cas7 puts through the RNA DNA hybrid here every 6 nucleotides. So this is a protein appendage that pushes through the nucleotides right there every 6 nucleotides. And you can see there's no base pairing at that position. And that's why having a mismatch at that position is completely irrelevant for the binding affinity of the complex. Okay, so that was known. It was really nice that we could see that in our data. We also saw a puckering caused by adjacent Cas7 subunits every 3 nucleotides. Now, this may be overinterpreting the structural resolution of this particular model, but we did see this consistently across different models. These subunits that line or the amino acids that line this pocket are conserved, and we tried to mutate them to try to affect this 3 nucleotide periodicity, but we could never purify cascade complexes. We think that all of this Cas7 RNA interactions, they're essential for cascade biogenesis to assemble that big complex. This is speculation, of course. But because of that, the protein pays a bit of a price in terms of its ability to bind CRISPR RNAs with very high specificity every 6 and 3 nucleotides. Right. So, with that in mind, what we learned was that Cascade can bind DNA very promiscuously. It binds off-target DNAs all the time, which is good because phages mutate to evade Cascade. But it's also bad because you can create autoimmunity. You can bind your own CRISPR locus, for example, or you can bind similar sequences in your own genome and then cause degradation of your own genome. So is there an additional mechanism to avoid DNA cleavage? Of course, I wouldn't be here if I didn't know what that mechanism was. And so here's what we did for that experiment. We took Cascade in one color and saturated the DNA clusters to the best of our ability with Cascade. Then we added Cas3 in a different color. And we just looked at the co-localization of Cascade and Cas3. Some clusters light up as white, meaning that Cas3 is there. Some clusters stay green, meaning Cascade is there, but Cas3 did not bind. I'll spare you the analysis and just give you the results. The results showed that Cas3 binding to a preformed Cascade RNA, CRISPR RNA complex is really sensitive to having mismatches in the positions, the first 3 positions of the PAM, -3, -2, -1 in my nomenclature, and then the first 3 positions of the CRISPR RNA complement. Okay, we validated all of that with experiments both in vitro and in vivo. Here's a gel shift showing as we change the number of mismatches and the PAM, you can actually see the Cascade Cas3 complex makes a really nice super shift here. But then what happens is the CSC1 subunit dissociates in a DNA sequence dependent manner. Okay, and without CSC1, I'm sorry, this is Cascade. This is Cascade plus Cas3, so that's the super shift. And if you play around with that sequence, you actually lose CSC1. And if you don't have CSC1, you can't bring in Cas3. So you have a subcomplex that forms at suboptimal DNA sequences. And does that matter for in vivo activity? Of course it does. If you can't recruit Cas3, you can't destroy, in this case, a target plasmid with a different antibiotic selection, okay, as you play with a target sequence. So all of this suggests that there is an additional mechanism by which Cascade can avoid recruiting Cas3 if it's at a suboptimal sequence, which is important for not destroying yourself. So now I'm going to switch gears in the remaining 15 minutes or so and tell you a little bit about our single molecule side of CRISPR-Cas adaptive immunity. Before I leave, though, I should mention that this using next-gen sequencing chips for massively parallel studies of protein nucleic acid interaction is really quite useful because it generates hypotheses that then we can test with more focused techniques, like you'll see in the single molecule side, or gels like this, right? We could never have stumbled across this unless we tried a lot of different sequences. And there's no reason to believe, in fact, we have some preliminary data to show that you can push the NGS to single molecule resolution. These are really nice flow cells. Illumina spent a lot of time making them quite high quality. All right, so back to the schematic of CRISPR-Cas adaptive immunity. I'm going to talk about primed acquisition now. I'm not going to talk about our work on naive acquisition, but I'll talk about primed acquisition. So what's all of this stuff doing together, and how does that work in primed acquisition? So for that, we use our DNA curtains approach. Here, using this approach, we can array thousands of individual DNA molecules here at lambda-phage DNA, the favorite DNA of molecular biophysics, where each of these DNA molecules is about 50,000 base pairs long. We can array them on the surface of a flow cell, which is fabricated to capture them in these nice little lines. This gives us a chance to see lots of different experiments. We can here extend them by buffer flow, and the DNAs are stained with a fluorescent dye. And buffer flow is used to keep them extended. Alternatively, we can capture both ends on little fabricated pedestals that we make in the clean room, and then turn off buffer flow, and the DNA remains taut like a guitar string. So for these experiments, we turned to our fluorescent cascade, and we just put it on the DNA molecule. So here what you're seeing is two fluorescent cascades running around looking for a target. This DNA molecule actually doesn't have a target for them, so they're going to keep running around looking for it. This is, of course, one-dimensional diffusion, and we see that for many, many different proteins that interact with DNA at specific target sites. To look at these images more quickly, I'm going to show you Kymographs. A Kymograph is a summary of that movie, where instead of particle tracking, we just take each single vertical slice represents one frame in the movie. So time is on the x-axis and DNA position is on the y-axis, and you're basically seeing that movie splayed out in a single image. So lots of proteins, maybe most proteins, that need to find a target on DNA, whether it's a structural target like mismatch repair complex, nucleotide excision repair complex, or a DNA sequence target. Lots of proteins scan DNA. Most of them do. Facilitated diffusion accelerates the target search. But most people don't really spend the time, so demonstrated that they don't really spend the time to understand, well, what are the molecular determinants or what are the molecular features that cause this facilitated diffusion? We decided to dive a little bit deeper into that. This is the CSE1 protein shown here. And we figured that there's this very strong channel of positive residues that are conserved across many, many different CSE1s. There's about 8 of them, generally speaking, and 5 of them in the cascade we're using. So that's that CSE1 that recognizes the PAM. They're sort of on the outer surface of this complex. And we thought perhaps these may be the structural basis for why Cascade can slide around on DNA looking for its targets. We made mutations. So we have 5. We converted them to alanines. And here you're seeing how long these trajectories last on average on non-target containing DNA. This is sort of a survival of how long Cascade can bind before it dissociates. Wild type under physiological salt can do so for 7 seconds or so. If you add additional charges, it increases only slightly. But if you take away those 5 charges, it doesn't go to 0. So there may be still additional ways Cascade can interact with DNA, but it significantly reduces the time Cascade spends on DNA. And this, of course, is borne out in a functional assay. Here we're looking at EMSAs again, and removing these 5 alanines that are not related to R-loop propagation or even PAM recognition. Structural basis for that is also known. That still kills the binding affinity 100-fold. And that's borne out in vivo interference assays as well. So this scanning is really quite important for target recognition and overall activity. And that's probably the structural basis for much of this sliding. So now we didn't want to keep Cascade frustrated. We actually added a target into our DNA. And here you see Cascade that did find a target, stuck around for a little while, left, which is surprising, and then continued on this match target. Okay, so that's how we analyze data like this. We particle track the individual cascades, and we look at how much time it spent on the target, walked away, came back, and so on. We see tons of these pausing events that don't convert into a completely locked complex. By the way, when Cascade makes a complete R loop, we measured this, the off rate is on the order of 2 days. It just doesn't come off, right? This RNA DNA base pairing is very tight. So as expected, Cascade binds its target and stays there after we let it scan around a little bit. But what was less expected was these, or surprising, were these excursions away from the target. If you analyze this for the perfectly matched CRISPR RNA matched target, it's a bi-exponential distribution of pause times with a short phase and a long phase, 3 seconds and 50 seconds. Looking at the details of that required making a second target. So what we did was we knocked in a second target into lambda that was partially matched to the CRISPR RNA and far away from the main target so that we could use the main target as sort of our control/internal standard. So we have 2 targets now, okay? And that's indicated here. So we have the perfectly matched target at position 21 kilobase from the left side, and we have a second target at 35 kilobases from the left side, and schematically showing what's changing in the second target. We either change the PAM or we change the amount of CRISPR RNA matched DNA here to the first third, second third, and last third. All right, so what do we see? We still see pausing. We see pausing at both sites now, except for when it's completely scrambled over here on the bottom in this histogram. And interestingly, we see that changing the PAM by and large changes the distribution of the fast component and completely abrogates the second component. Okay. Whereas from a good PAM to a medium PAM to a non-permissive PAM. Whereas changing the CRISPR RNA matching part from the first third, second third to third third, what we found was that there's still this 3-second component, but the amplitude of the second component goes down. And interestingly, it goes down for the middle segment, but not for the last segment. This was surprising because, well, we thought that R loop propagation should start from the PAM proximal side and proceed sequentially to the PAM distal side. We have seen this. A single molecule Fred paper from Chirleman Zhu's lab has seen this. I'm not quite sure what to make of it. I'm going to give you my speculative answer of why a PAM distal match can somehow still cause Cascade to pause. Okay. So this comes from work from Ailong Ti's lab, who is my collaborator and has solved the high resolution structure of a partial R loop complex from this exact Cascade. What they see is that the CSC2 subunit, which I haven't talked about yet, has 2 salt bridges. These salt bridges can engage after CRISPR RNA is formed. Okay. So we think of these as intermediate locks on this CRISPR RNA. And to cause this transient pausing, you can actually, to cause complete binding that essentially complex can't fall off, both salt bridges have to form. This is called this locking, R-loop locking mechanism first proposed by Ralph Seidel's lab based on magnetic tweezers work. However, these salt bridges are semi-independent. These are 2 different subunits, and they can lock independently of each other. So if you have partial matches, the CRISPR RNA can actually run through a few mismatches. We saw that in our high throughput work, and lock one or both of these salt bridges. If only one of them locks, then you get a short pause. If both of them lock, you get a long pause. So we haven't done the mutagenesis to prove this exactly, although Ailong has the constructs, but that's currently our working idea of why you can get longer pauses on these partial matched substrates. All right, so in the last few minutes, I really want to get to Cas3, how it functions with Cascade, and what I'm going to call the primed acquisition or what we called the prime acquisition complex, which is of course the ensemble of Cas1, 2, 3, and Cascade. So switching topics to Cas3 now. Cas3 is a motor protein. Okay. We wanted to study how Cas3 works. Cas3 is loaded onto DNA by Cascade. It cannot bind DNA on its own. Okay. And Cascade has to be R-loop, has to have formed its R-loop to load Cas3. So we used a poor man's version of force control. We put a big bead on our DNA curtains and used buffer flow to essentially cause a big drag on that bead and cause the DNA to extend as a function of force. We can use the worm-like chain model to calibrate that, and we can exert up to 20 piconewtons of force onto our DNA molecules before we rip the DNA molecule off the surface. All right, so what does the data look like? So here we have Cascade in magenta and Cas3 in green. The target is labeled as T. The barrier is shown on top, and the F stands for free end, or I didn't want to use B again for bead, but this is the free DNA end that has a bead on it. All right, so under moderate force, what we see is that Cascade and Cas3 start moving together once ATP is added, okay? And at some point, Cascade snaps back to its target site. Okay, so in fact, we don't see the DNA is completely degraded. So that's about half the time. And about half the time, we see Cascade actually stays target site bound, and Cas3 just takes off, right, independently. Now, the only way this could happen is if Cascade loads Cas3, and then Cascade, Cas3 start moving together, forming a loop of double-stranded DNA, or partially single-stranded DNA. We think it's mostly double-stranded with a bunch of nicks in it. I'm not going to show you that data. And then as a function of force or stochastically, sometimes the Cas3 Cascade interaction ruptures. Cascade, which is still got an R loop with the target DNA, snaps back to its starting position, to the target site, and Cas3 continues on its merry way, right? So this is indeed true. We can increase the force, and we can actually see that we can cause this loop rupture, the lower of the two events, to occur more frequently when Cas3 is being pulled on, when the whole complex is being pulled on by this bead. All right, so 2 more slides before I wrap up. So then we wanted to, and this is a very processive complex, we wondered what happens when these Cascade Cas3 complexes start running into roadblocks. So we engineered a number of roadblocks. Here are some EcoR1, catalytically inactive EcoR1s that we can deposit onto the DNA on one of 4 sites. The question was, what happens when Cascade Cas3 runs into a roadblock like EcoR1? We also looked at lag repressor and RNA polymerase, E. coli RNA polymerase. So another DNA binding protein, a transcription factor, and RNA polymerase. These are all likely to be encountered by E. coli Cascade in E. coli. We see a number of events, including stalling of the moving complex. So here the roadblock is now in green. Stalling of the moving complex, sometimes movement of the complex, and sometimes, this was the coolest one on the bottom, relooping. So it collides, falls back to its target site, collides, falls back to its target site, and so on. This is quantified here. But if you look at the processivity of the Cascade Cas3 complex, it's very processive on naked DNA, shown here in this B-swarm plot, 20 kilobases or so. The diamond indicates the median. But as soon as you put a roadblock, it's quite obvious that if you look at the first collision event, EcoR1 at the first site, Cascade stops there. Second site, Cascade stops there slightly earlier, and lag repressor, same thing. Right. So really what it says is that in a majority of the cases, a single roadblock is sufficient to block Cascade Cas3, and only infrequently does this roadblock get evicted by Cascade Cas3. Final data slide. I'll tell you about the whole primed acquisition complex, which was a really cool surprise from all of this work. Then we wanted to ask, well, how does Cas1, Cas2 use Cascade and Cas3 to acquire memory? Okay, Cas1, 2 is a DNA binding protein on its own, doesn't have target specificity, very strong target specificity, I should say. It binds DNA very transiently on the order of a few seconds. When you add Cascade, however,
2 more minutes.
Perfect. When you add Cascade at the target site, that it recruits and stabilizes Cas1, Cas2, an interaction that had not been seen previously. Okay, so Cas1, 2 here on the bottom co-localizes nicely with target-bound Cascade and stays around a long time. Now, if you add Cas3 to the whole mix, now this is about a megadalton complex that we've assembled here. When you add, because Cas1, Cas2, as you can see, it's a tetramer and a dimer as well. So it's a 6 subunit complex. When you add Cas3, the entire thing takes off. So Cas3 here is not fluorescently labeled. We could have done a 3-color experiment, we didn't, but you can see Cas3 activity because it's got an ATP-dependent translocation, so the whole complex moves. Cas1 moves, Cascade moves, and Cas3 moves, and it moves for a very long distance. So what this says is that the acquisition machinery can actually interact with interference machinery. And I'll speculate about why that's important. Genetic experiments have shown that primed acquisition can occur tens of kilobases away from a priming site. How does that work when we know that a single roadblock can block Cascade and Cas3? Clearly, this complex won't move 10 kilobases without hitting something, right? So here's a quantification of the lifetime of Cas1, 2 on DNA. You can see on its own, it's very short-lived. Add Cascade, it stabilizes, and that's magenta. If you assemble what we call the primed acquisition complex, which includes Cascade, Cas1, 2, and Cas3, it becomes very long-lived. So together, this complex is stable. By the way, there's no structural data on what this complex looks like yet, and our collaborators are working hard on getting this whole complex up together no primed acquisition complex has been structurally solved. But what's really cool is that once you assemble the whole complex, in fact, it becomes much, much better at getting through roadblocks. Okay, so comparing the Cascade Cas3 complex against the primed acquisition complex on the left bars, you can see that 100% of them maintain this loop. Okay, and then of the ones that collide, and I guess I haven't shown that data, but of the ones that collide with a roadblock, the roadblock is rapidly removed by this whole primed acquisition complex. So the whole complex is very stable and is stable against roadblocks. So it can translocate for tens of kilobases without falling off or getting stuck in front of an ECOR1 or an RNA polymerase. So putting all of this together, here's our model of how primed acquisition works. Okay, you've got Cascade scanning around on DNA, finds a PAM, opens an R loop, propagates the R loop. This requires the activity of CSC1 and its nonspecific DNA binding cleft. Okay, then primed acquisition also requires Cas1-2. We have shown, I didn't show you this, but we have shown in the paper that Cas1-2 can interact with Cascade, or first Cas3 can interact with Cascade. If Cas3 interacts with Cascade, then you got interference. But this subcomplex can then assemble into a primed acquisition complex. During primed acquisition, Cas3 probably nicks the DNA. Cas1, Cas2 recognizes PAM-like sequences and is also a nickase of its own. Okay, so together, Cas1-2 is just sitting there picking up pieces of DNA that are adjacent to this whole complex. And eventually, we think Cas1-2 will dissociate with a short piece of DNA that it excises from this loop and then inserts it into the adjacent to the leader's side of the CRISPR locus. There is an alternative pathway shown by several labs, including at Weizmann, Rotem Sorek's lab at Weizmann, where the host nuclease RegBCD can also target the foreign DNA, producing short oligo fragments, which then only require Cas1-2 to be integrated into the CRISPR locus. And this whole interaction between Cascade and Cas3 and Cas1-2 is also seen in Cas9 system. Cas9 also has a Cas1-2 associated with it and additional proteins of unknown function. But for primed acquisition in the Cas9 system in Streptococcus pyogenes, shown by the Marafini lab, Cas9 is also genetically required for this activity. So probably something similar is happening. With that, I'm going to conclude. I'd love to get some structural insights into either what this looks like, the primed acquisition complex, or what the Cas9 primed acquisition complex looks like. All of that is still unsolved. So Act 1 was our hacking of Illumina sequencers, which can really increase your throughput for protein nucleic acid interaction studies. And Act 2 was our use of some of those hypotheses generated in the high throughput study to really go after the mechanisms at high resolution using single molecule approaches. I'm going to stop here and thank my lab and remind folks here that Austin is a good place to live. And if you are interested in any of this work or want to hear about some of our other what's cooking in the lab, please email me and I'm happy to talk to you about job openings. So I'll stop here.
Okay, thank you very much, Dr. Finkelstein, for the very nice presentation. We do have questions in the chat. I'll read out. John Tanner asked, I assume that 1D diffusion is bidirectional, so it goes 2 steps forward to 3 steps by type of scanning.
Is that a question or more of a comment?
That's a question. Well, I guess it's more of a comment, but I guess if you have any sort of insight on that, that could.
Yeah. So of course it is bidirectional. One-dimensional diffusion is bidirectional. My belief now is that most DNA binding proteins are doing some sort of one-dimensional diffusion. Streptococcus pyogenes Cas9 does it as well. That was shown by single molecule FRET studies. It just does it on a smaller length scale. Cascade has been imaged within E. coli cells, and it also has been shown to do this in live E. coli.
So that also answers Dr. Tanner's another question, whether it's directionality. Now, Dr. Honghua has this question. She said, Ilya, great talk, very impressive work. for Cas3 translocation by looping DNA, do you know how long is a DNA loop?
We know that the loop has to be, in some cases, up to 20 kilobases long because Cascade is always target bound. So we've never seen an example where Cascade leaves its target. That CRISPR RNA really base pairs with the target DNA. But we've seen translocation on the order of 20 kilobases or so. So that DNA has got to be going somewhere. Yeah. So there's been analogous so that's a really large loop. There's been analogous examples actually in the REC-BCD system. Stephen Kowalczkowski's lab, Maria Spies, who was his postdoc at the time, showed that the REC-BCD complex has two motors, REC-B and REC-D. These motors travel on single-stranded DNA with opposite polarity, but also different speeds. The speeds differ by a factor of 3. So one motor is ahead of the other motor, and that causes a single-stranded DNA loop to occur, right? And they could probe that with P1 nuclease, for example. And that has really interesting implication for homologous recombination. In this case, we don't have two motors. We have one motor, but we also have a stationary anchor. I should mention, we see something like this in our cohesin work as well. So we recently had a paper showing that human cohesin is a DNA translocase using similar approaches. And in follow-up work that I haven't yet talked about because it's almost done, we've also reconstituted the CTCF dependent stopping of cohesin, and we can see one directional looping where CTCF cohesin, we think cohesin moves as a dimer. One cohesin is stuck at a CTCF bound site, and another cohesin continues to loop extrude. So this happens a lot, I think, and these loops can be quite dramatic.
Okay, great. I think the time is up. We should move to the next talk. And thank you again for the wonderful talk.
I look forward to answering questions on the platform, which I'll be jumping on right now.
I'm pretty sure there will be quite a few, and people will probably contact, even now you're in our group, and they'll maybe reach out to you privately. Okay.
Absolutely.
Thank you
for your attention.
Mm-hmm. Okay, our next talk is going to be given by Dr. Vasenci Jayaraman, who's a professor in my university, University of Texas Health Science Center at Houston. And Dr. Vasenci has been working on the ionotropic glutamate receptors for many, many years. And she's been focusing on single molecular studies of these amazing ion channels. So she's going to tell us about the glutamate receptors from conformation to function. Vasenci, take it over.
Yes, thank you, Mike. And thank you for inviting me to give a talk about our work on glutamate receptors. I'll start off by thanking the people who did the work. So today I'm going to be talking about one specific subtype, Amperoceptives. And this work was done by Elissa and Sana and Vlado, who are all senior postdocs and senior scientists in the lab. And I won't be talking about the other subtypes, NMDA, where both Nidhi, a postdoc, and graduate student Ryan work on it, or K8 subtype, where Nabina is working on it, and Doug, who just graduated, is working on it. So let me get started by giving you an introduction of what glutamate receptors are and where they are present and what they do. The ionotropic glutamate receptors are present at the synapse, as you can see here, and they are in the postsynapse and receive glutamate molecules as the signal. When glutamate binds to the ionotropic glutamate receptors, they open cation channels. And the AMPA subtype of the ionotropic glutamate receptor is involved in very fast excitatory signaling. So what it does is it opens, sodium ions go in. And so when sodium ions go in, you see channel, the currents going inward because of the fact that the ions are going in. And then this receptor eventually desensitizes, and it desensitizes very quickly, as you can see. And that's characteristic of Amperoceptives. So my lab has been focusing on understanding what the conformational landscape is that is associated with this activation and with the desensitization process. So why are we interested in this protein? They play a critical role in learning and memory because they act as the on switches in the synapse, and that's why because they're excitatory. And mutations in the protein are known to cause a deficiency in function, and or deficiency in function are known to cause learning deficit, as well as overstimulation causes epilepsy. And also oxygen deprivation leads to glutamate accumulation at the synapse and causes overstimulation of these receptors, and is thought to be involved in the neuronal death in stroke. So getting onto the main question of the mechanism of activation and desensitization, I'll first start with the mechanism of activation of amperoceptives. So again, what we are looking at is this process of channel opening. So Amperoceptives, the structures have been very well determined. It is a dimer of dimers. There is amino terminal domain. In the Amperoceptives, the role of the amino terminal domain is not established clearly, but it's thought to maybe interact with presynaptic components and adhesive proteins that connect the presynapse to the postsynapse. But in case of NMDA receptors, for instance, this is the place where modulators bind, such as zinc ions and so on. The main action part is here, which is basically where the agonists bind, and therefore it's called the agonist binding domain. And then once the agonist binds, then the channel that initiates the conformational changes, and this is the channel part where the ions go through. So that's the transmembrane segments. So we first started on working on this a while ago, and I just wanted to bring this up mainly to sort of familiarize you with the different techniques we use in the lab. This was a modified luminescence resonance energy transfer method that Jen Gonzalez, who was a postdoc then, started and initiated this project. So what we do is basically, fortunately for us, Amperoceptives don't have too many non-disulfide bonded cysteines. So in this case, we just use cysteines as a mechanism to attach donor and acceptor fluorophores. Of course, right now, the technology to attach donor and acceptor fluorophores has really exploded. In other words, we have DNAs that can be used to introduce unnatural amino acids at whatever position you want. And we do use unnatural amino acids for specific labeling too. But cysteines are much easier, and so we remove the three non-disulfide bonded cysteines in the extracellular domain, and then we introduce cysteines wherever we want to attach the donor and acceptor fluorophores. But we want to do this in cells. So this particular method is, we want to see what are the conformational changes of glutamate receptors in cells, or amperoceptives in this case, in cells. So in order to do that and to remove the background, the way we do that is we introduce a protease cleavage site between the donor and the acceptor fluorophores. So then what we can do is basically measure the lifetime. In this case, we're measuring the sensitized lifetime of the acceptor fluorophore, because the donor is a terbium chelate, which is actually luminescent and therefore has very long lifetimes. So it really helps us in being able to get good lifetimes for the acceptor fluorophore when it takes the energy from the donor fluorophore. So this is the lifetime that you have before adding protease to the cells. So these cells are just in cuvettes, and then we just measure the lifetime. Then we add a protease to the cuvette, and it cleaves off, as you can see, one site because it's between the two sites. And therefore, whatever is left is the background. So any cysteines from other proteins that are in the cells that have been labeled will give rise to this background. In this particular experiment, we were fortunate, we had very good expression, and so you can see the lifetime from the background is very less. But the reason we needed to do this was because there were times when there was not that good expression, there was variability in expression. So this really allows us to quantify the background and remove it. So throughout my talk, I'll be switching between the different techniques. Whenever we do LRAT, it's done in cells. So it gives us a more physiological context, but it's average FRET measurements. And then I'll be talking about single molecule experiments that we've done where we pull down the receptor, and that's not in the context of cells, it's individual molecules. Okay, so this is again done in cells. And so we looked at the conformational change across the agonist binding domain. The agonist sort of binds in the cleft between the domains. And what we see is the apostate has a slower lifetime compared to the glutamate bound state. And of course, from the lifetimes, we can measure distances. And so we know that the distance across the cleft is longer in the apo state versus in the glutamate bound state. And this was a while ago, and later on we've had structures come up. And so what we predicted is sort of there in the cryo-EM structures, where you see the agonist binds, and when the agonist binds, the cleft closes, and the cleft closing causes the channel to open. So again, the agonist binding domain is right here. The agonist binds, the cleft closes, and the channel opens. So this was work done by the Guo lab as well as Sasha Sobolowski's lab in Columbia. Both of them had very similar structures and very similar mechanisms. So the question, of course, next was what causes partial agonism? So we know that the cleft closes and the channel opens and you get currents, but then you can block the desensitization that I showed you by using drugs to stop desensitization. So then you're able to measure the maximum currents really well. And what you see is there are different agonists with different efficacies. So the question is, what is the underlying mechanism for some agonists giving larger currents and being full agonists, while others give smaller currents and are partial agonists? And this was actually predicted even early on by the isolated domain crystal structures, where they showed that different partial agonists with lower currents had smaller cleft closure, whereas those with full agonists like glutamate had larger cleft closure. As you can see, it's more closed down here. So we then said, okay, let's make sure this is the case in the full length receptor, and it followed the same trends where we measured lifetimes in cells, and we saw the same trends where extent of cleft closure as measured by distance across the agonist binding cleft really correlated well with the increasing activation. And sometimes it's best to just leave things as is and move on to the next project. But as scientists, we want to know everything about these proteins. And so we said, let's make mutations, let's do more experiments. And when we started doing that, things started to fall off the line, as you can see. And in fact, Eric Go's lab also crystallized this in collaboration with Jim Howe in Yale, crystallized this construct. And again, even the crystal structure showed a more closed cleft, but single channel level activation showed that it was only partially activated. So why are these not falling in line? We know it's not because of the fact that these were structures of isolated agonist binding domain, because our experiments in FRET were done on the, in LRAT, were done on the full length receptor in cells, so that's not the reason. So then we sat down and thought about it and said, well, that's because in structures, we tend, especially because at that time it was primarily X-ray structures that were used, you tend to crystallize in a given conformation. And for the LRAT measurements that we were doing, it's ensemble-based measurements, right? So so it gives us only an average. So we realized what we were missing was understanding all the states that the protein was occupying, for which we had to do single molecule measurements. So Anu, who was a graduate student at that time, started single molecule measurements. And the way we do it is basically we do pull down. So we have a slide on which we have biotin and streptavidin. And so then our protein has a streptavidin tag, and then we just pull down our protein onto so we express our protein in HEK cells, and then we pull down our protein onto the slide, and then we do the measurement. So it's really no purification prior to putting it on the slide. So the slide is in situ pull down, and the protein is just taken from the membrane preps that we have. And then of course we do confocal imaging. Of course you can do turf imaging too, but this is just confocal in our lab. And we measure the intensity as a function of time for the donor and the acceptor. And what you're seeing here is basically the acceptor intensity is high. So this is showing the FRET signal where the donor is able to transfer its intensity to the acceptor. The acceptor then photobleaches. and then the donor goes up. So this is sort of what we are looking for, making sure we have single photobleaching steps so that we are looking at a single distance. So we underlabel our receptors so that and make sure that the sites are such that there is no crosstalk between the subunits. And the fact that the glutamate receptors is very large makes it very easy for us to find sites that don't talk to each other across subunits. And we're looking for the single photobleaching step so we know that we are measuring one distance. And from this, if this is the fretting region, so in this region, we know how much fret is happening and we plot the fret efficiency. And in this particular trace, the protein is just in one state. It's not fluctuating between states. The green is the observed data raw from the microscope with no data manipulation being done. And the black is the denoised data. And you can use different kinds of denoising. We use wavelet-based denoising that Christy Landis at Rice her lab has developed. And, but you can pretty much use any other kind of denoising because people from other labs use other denoising. So here you have the observed, and in all of my data, most of the, towards the later part, you'll see we have the gray as the observed and the red as the denoised. But initially we just showed just the observed or the denoised. So this is just sort of telling you single molecule histograms, okay? So the molecule can either be in one state or it can switch between states as you're seeing here. And that sort of tells you how many states the protein is exploring. And from an ensemble of all those different histograms, we can then look and see how many states the protein is exploring, what is the shape of the histogram. And in this case, you can see for the Amperoceptives, again, we are measuring across the cleft. You see that the apostate is more open, lower fret, more open than the glutamate bound state. But it's more than that, right? This is the beauty of single molecule fret in that you can actually see the profile of this. You see that it's broad and sort of symmetric in some cases, but not in these cases. So this actually matched beautifully the data that we had from, not the data that we had, the computational work that Benoit Roux's lab had done. And you can see that the apostate is broad and the glutamate bound state is deep. This is free energy calculations, so it's not a direct correlation to distance, but it just tells you the profile. The profile is broad for apostate like we had, and narrow for glutamate bound state. So again, all of this is great. Everybody is happy. The computational guys, their work agreed with our single molecule work. The exact distance changes agreed well with the crystal structures. The average that we measured with LRAD agreed with the average of the SM fret. So really everything could be accounted for because we were looking at the complete conformational landscape, right? But the question was, why were these mutants different? Why were they not following the trend? So we picked this mutant T686S, as I said. And when we did the single molecule fret, it became obvious as to why we're not agreeing. The crystal structures were more closed, and the most probable state here is more closed. But you see that the protein is sort of moving around through different structures, and it's not staying in this closed cleft state, which is what leads to activation. So since the protein is exploring all these non-functional states, it's therefore the activation is lower. So we said, what if we, instead of talking about a specific state, we say, well, what fraction of the protein is in this closed cleft state? So when we did that, then we saw all the protein, all the mutations, all the ligands with different activities, they all fell in line. So what this really tells us is that it's not one state and it's not an average. The best way to look at this is to say, what is the fraction of the protein that's in this active state? And when you're able to characterize that, then the function and the conformation are very well correlated. So it's great that we were able to look at the protein by itself. The next question was, can we start looking at the protein in complex with other auxiliary proteins? In this case, we looked at gamma-2, which is in the presence of which, this is an auxiliary protein that's present in the brain. Pretty much amperoceptors are always decorated by auxiliary proteins in the brain, and they change the function. So really, we need to understand, does this mechanism of cleft closure hold in the presence of these auxiliary proteins? And in particular here, what you're seeing is that kainate is becoming more active in the presence of these auxiliary proteins. And therefore we started looking and saying, first, let's do the LRAT measurements, see the average and see where it's going. And what we saw is basically, this is in the case of the Amperoceptives without auxiliary protein. And this is the Amperoceptive with the auxiliary protein. And you can see that even in the apo state, it's already smaller lifetime, which means the cleft is already primed. It's already ready to be closed. And then when you have kainate, it quickly goes to the full closure state and therefore is able to activate it as much as, say, glutamate, for instance, in the absence of gamma-2, which is what we were seeing. So based on this, we said, so the trends are the same. So the mechanism is very similar. It's just that the protein is already primed to be activated and therefore activates much better in the presence of the auxiliary subunit. So we said probably the auxiliary subunit, the gamma-2, is sitting under the cleft, the agonist binding cleft, already propping it closed, so that when the agonists bind, it only explores the closed states and doesn't explore the open states as much. So in order to see if that is true, what we did is we put a natural amino acid at different positions. And this is just a schematic of how we do a natural amino acid. Is very similar to what everybody else does. We don't design the tRNAs and RNA synthetase and so on. We just get it and then we just use it. So it's pretty straightforward. And in this case, our unnatural amino acid was benzoyl phenylalanine, because it can be used to cross-link. And so we put that in at different positions, and you can see that there are specific positions where it cross-links to the gamma-2, not in all positions. And based on sort of this Western blotting where we see where it can cross-link to gamma-2, we sort of predicted where the sites were, and sort of these were the sites where it was binding to gamma-2. And since we know where the sites are, we said, it agrees with our hypothesis that gamma-2 is sitting under the agonist binding domain, causing the cleft to only explore more closed states. And then what we did was LRAT measurements, sort of nano positioning measurements. And we put 3 unnatural amino acids on the gamma-2 and 1 cysteine on the amperoceptors, and measured distances between the cysteine and the unnatural amino acids. And based on the distance, it's like a GPS kind of system where there is only one point where all 3 distances are meeting up. And so using that as the docking point, we predicted this to be the structure. Remember, all of these are done in cells without requiring purification. So this is the sort of structure that we predicted for gamma-2 binding to amperoceptors in cells, and the cryo-EM structure of the isolated protein in the presence of gamma-2 showed very similar structure. So, in terms of gamma-2, we were able to show that the reason that gamma-2 is able to activate better is because it favors the closed cleft conformation. It's priming the receptor so that it's ready for activation. Then the next question is that desensitization that I haven't been talking about up to now. It's this part, when you don't block activation, when you don't block desensitization, the protein actually quickly desensitizes in the presence of glutamate. So what is the mechanism underlying this desensitization process? So even early on, even before these cryo-EM structures were done, the hypothesis was, since the cleft is closing, there is a strain on the transmembrane segments. And this strain, because of the strain, what they expected was that the dimer interface between these agonist binding domain gives up, causing desensitization. So when the dimer interface gives up, then the channel segments collapse and the channel is now closed, right? So these are the different structures, and you can see clearly that in fact, different classes, different, there's very, very large decoupling, very small decoupling in the cryo-EM, whereas the crystal structure showed no decoupling.
Essentially.
Yeah. So based on this, so there was this thing is like, how much decoupling do you need for the protein to undergo desensitization? So we did single molecule FRET because we can look at all the states that the protein explores. And here we are measuring decoupling at the amino terminal domain, and here we are measuring decoupling at the agonist binding domain. And what you see is, in fact, yes, the glutamate bound form at both the amino terminal domain and the agonist binding domain is exploring different forms, but it tends to populate certain forms. And based on these distances, we said that most likely class 2 is the most common form that is leading to desensitization. So now again, we can correlate structure to conformation to function. The final part is how does a modulation by another protein, which is gamma 8, which actually doesn't allow it to desensitize, what is happening in that case? And we can compare this to what is happening in the case of cyclothiazide, a drug that blocks desensitization. And what we found is, doing single molecule FRET, that it's very similar to cyclothiazide, except that the amount is not as much, and therefore you saw in the protein, in the function, that it's not as much as here. So we can pretty much see which conformation corresponds to which function. This is the open, and that's the desensitized. It's more open with cyclothiazide, less open, but still open in the presence of gamma 8, whereas here it's completely desensitized. So again, same thing in the agonist binding domain, we could correlate that this is very similar to CTZ, except we're starting to populate more of the decoupled state with gamma 8. We can actually start to compare what we are seeing in single molecule FRET to single channel recordings, because these are recordings from individual channels. And what we found was the lifetime for these states really quite well match the lifetimes that we are seeing in the single channel recording. So really we can start making comparison between conformation at the single molecule level to function at the single channel level. And based on this, we can start thinking about how can we inhibit specific states. So then we went on to start, this is just to show that the conformations agree, but then we can say, okay, if this is the conformation, can we use drugs to specifically inhibit? And that's what we did here, where we showed that zinc inhibition can happen better in certain conditions versus others based on our conformational data. This is important because there are certain kinds of editing that are involved in ALS. So maybe zinc itself could be used as a drug to sort of at least slow down the progression of ALS. So I want to end with that, saying how we were able to correlate specific conformations to specific functional states because of the fact that we are able to do single molecule FRET as well as single channel recording. And end with, of course, the acknowledgement, which I already did. So I'm ready for questions.
Well, thank you very much, Vasanthi, for a very interesting talk. Are there any questions in the audience? Please enter in the chat in Brahma. And of course, if you're trainees, you can put a note and say, I'm a graduate student, your question will be prioritized. I guess I have a question. I find the very last part is very interesting, where you're comparing the single molecular changes and the single channel activity. But I guess technically it would still be very challenging to actually do that simultaneously, right? So those traces are from different measurements.
Absolutely, absolutely. They are done on two different instruments on two different. So yes, they are two different. But so at this point, all we are able to do is compare the kinetics. And again, the kinetics I want to stress is we have been able to go down to a few milliseconds, 5 milliseconds. And maybe if we push the thing, we can go to 1 or 2 milliseconds, but not faster than that. And it works for that system, even though I said Amperoceptives are very fast. Remember, gamma 8 stabilizes it in the open channel state. So it tends to be in bursts. So the burst lifetime is what we are able to compare. So the limitation is we're not able to compare fast kinetics. So it's only the longer kinetics. And the longer kinetics in this case correspond to that specific conformational change. So it works really well if you're looking at milliseconds or longer, and you don't want it to be too long either, because the photo bleaching will happen in a few seconds to minutes sometimes. So it can't be too long. So it has to be in the sweet spot where you're able to do that kind of comparison. And at this point, it's on 2 different instruments, so it's just comparing kinetics.
Mm-hmm. It would be very hard to, for the most part. Have you tried this in the neurons? I guess in the neurons, it's always been very difficult to measure single channel activity Amperoceptives.
Right, and it's very difficult for these glutamate receptors because they tend to cluster in the neurons, right? So it's very hard to do single molecule FRET for these specific proteins in neurons, because they tend to be clustered. So you're not going to have single molecules at any given. We've tried it, but even if we express it at very, very low concentrations, they tend to form clusters. It's always more than 1 photo bleaching step. So yeah.
Okay, great.
Maybe other proteins, yeah.
Yeah, are there additional questions? Well, if not, we thank Dr. Vasenci for a wonderful presentation. Very detailed studies on the glutamate receptors, dynamics and everything. Very exciting.
Thank you.
Okay, thank you. Okay, so we now move to the trainees talk, and we have Abigail Koski, who's a graduate student from Rice University. She's in her fourth year of graduate school, and in the Department of Biosciences in Rice University, and her interest being in the field of crystallography and cryo-EM. And she's going to tell us about the structure interrogation enzymes involved in biosynthesis of anodyne natural compounds. So Abigail, you have the floor.
Thank you very much. I am going to share my presentation. Okay. So as it has been introduced, my name is Abigail Koske. I am from Kenya, and I'm doing my graduate studies here at Rice University, being a 4th year at the Phillips Lab. And my research is focused on interrogation of these enzymes that are involved in the synthesis of natural products, and specifically in dynes. So just to begin with, natural products has been really a big source of small molecule therapeutics in the drug industry. And we have been able to harness so many molecules from these natural products in the past, although still we have more potential in them. And there are a couple sources of natural products in our lab. We focus on bacteria, specifically compounds from Micromonospora, Echinospora, and we have seen, and it has been shown in the past, that we have a couple compounds like tetracycline, chloramphenicol that we use day in, day out in our labs when we are doing experiments. So here, one of the natural products, or the natural product that I'm focusing on, is the indines. And just to introduce what indines are, these are complex molecules that I will show in the next slide. The indines were first discovered in the 1980s, and they're metabolites derived from actinomycetes, naturally found in the soil. Actually, why we are interested in indines is that they have an ability to cause cell apoptosis by cleaving DNA, either single-stranded or double-stranded. I'll show you in the following slides, but here is an example of a drug called Myotag that was initially introduced in 2010 and then taken out of the market, but then it was reintroduced in 2017 with a different drug regimen. So here, this Myotag is involved in treating amylogenous leukemia. So the indines have this unique molecular architecture as shown here. These are just examples of a few indines, 10-membered ring indines. And in red is the indine core that is made of 2 acetylenic groups and conjugated by a double bond. And to color the indine core and the peripheral moieties that play a couple roles. So the indine core is the one that is involved in the DNA scission, and that's why it's called the warhead. And the rest of the peripheral moieties will help with locking it or directing it directly to the DNA. And if you administer it to the body, like any cell, it will cleave any DNA. So we want to try and ensure that we can directly or specifically target a certain DNA. And so in the past, been able to do that by conjugating it with an antibody to be able to direct it to the specific tumor cell. So the indines actually are, they cause DNA cleavage by, first of all, they undergo this Bergmann cycloaromatization to form this active diradical, which is capable of abstracting hydrogen on the DNA, causing either double bond DNA cleavage or single-stranded DNA cleavage. So just to simplify how so we're interested in looking at how these complex compounds are formed, and just to break it down to what we think is they are formed. Initially, you form a linear polyene in the presence of a starter unit and an extender unit and NADPH to form this linear polyene called a heptaene that I will mention later. After that, it undergoes cycloaromatization in the presence of 9-membered or 10-membered ring, specific associated coenzymes to form the core. And then in the presence of the enzymes associated with forming the peripheral moieties, it forms the different 10 or 9-membered indines. So we could decide to synthesize it from scratch, a total synthesis of this indine compounds, but it's proven challenging in the past years. And one of our professors in Rice University has been hands-on involved in total synthesis of this indines. And one of the examples is calicheamycin. Here, it took 5 years to synthesize the whole of calicheamycin in 24 steps, but we only get a very small yield. So the study of the biosynthetic gene cluster has helped to be able to synthesize this genes and learn what each gene in the biosynthetic gene cluster does to be able to assemble these complex molecules. But looking at the biosynthetic gene cluster of calicheamycins, for instance, we have about 80 genes that are involved in the biosynthesis of calicheamycin. Here, the gene in the biosynthetic gene cluster, the different genes have been color-coded to show what it does. Within the several genes that we have here, there are some with the unknown functions. And one of the, there being 80 genes that are involved in the synthesis of the calicheamycin, then you can think, so how long, how long will we be able to be able to learn what each gene does and how they play a role in assembling these complex molecules? And well, a couple other structures have been solved already using X-ray crystallography, and there are different structural methods out there. And the role I could play is probably solve one of them. And as we have calicheamycin and other indines, they play almost the same roles. So therefore that is an advantage on my side. And in our lab, or like in this talk, I'm just gonna mention the study of two of the genes involved. One is involved in the biosynthesis of calicheamycin, and it's called CALU17. And the other one is involved in the biosynthesis of dyneamycin, which is called DynF. So looking at the structures of calicheamycin and dyneamycin, you still see the indine core, which is conserved, the indine core, and then that difference is the peripheromoides, the coloration. You see that you have a couple functional groups in that we're trying to understand how do those genes involved in the biosynthetic gene cluster enable the assembly of this whole complex molecule. So I use X-ray crystallography to be able to determine the structure of CALU17 and DynF. And I'm gonna show you the structure of CALU17 and DynF. CALU17 structure was solved to about 2.5 angstroms. And within that structure, there's something like—
to leave some time for question, are you going to be able to wrap up in a couple of minutes?
Yes, sir.
Okay,
yes.
Thanks.
So the CALU17 has a catalytic cleft that we hope to decipher what it does, actually, the function of CALU17, the biosynthetic gene cluster of calicheamycin. And also for DynF, we were able to solve the structure to about 1.5 angstroms. And within the structure, there's this palmitic acid that might give us a clue to what DynF does. As I mentioned earlier, DynF and CALU17 they were proteins with a known function. So we hope that by solving the structures, we'll be able to determine or characterize what it does in the biosynthetic gene clusters, and also to be able to give us or inform us as to the drugs that we could be able to develop in the end. And so I'm just wrapping up with the conclusion that with the solving of structures of these two, DynF and CALU17, it would help us to determine the roles of this enzyme within that biosynthetic gene cluster, and henceforth be able to come up with drugs for anti-tumor drugs for in the future. So, and I'll just like to acknowledge my lab members, our collaborators at the University of Kentucky, the NACPRO, and then our grants. Yeah.
Well, thank you very much, Abigail, for a very, very nice presentation and very challenging work, I assume. Are there questions from the audience? Well, I guess I'll begin with, you're basically talking about a group of a natural compound. Is it, are they, how commonly are they found in the nature, with the abadan-based chemicals or compounds?
How common are they found?
Yeah, how common are these?
So they're naturally found in the soil. And so just the organisms that make them a Micromonospora, Echinospora, are found in the soil. So with that, when it was first found, we just wanted to know, like, how do they actually make these compounds? So, and with that, we could be able to synthesize them and to be able to use them to develop novel drugs eventually.
Yeah, I am still trying to imagine, from say the natural enzymes that you have crystallized and understanding the structure. How are you going to utilize that information and for chemical synthesis? I'm not a chemist, so I'm not really.
Yeah. So first of all, as I mentioned, there are a couple of genes involved in the biosynthesis of these complex molecules. So it will be one. So we're trying to build a library of all of them. Once we solve all the structures, then probably we'll be able to find a way of coming up with a chemical. Yeah.
Okay, great. Thank you very much. Well, I also like to thank the three speakers of our session, for this first session, and also the first practice using Umbrella, for the meeting platform. It's been working so far so well. And also like to thank the active participant, our audience, by entering your questions in the chat. I encourage them to continue to do so, and especially the graduate student and the postdocs. You can enter your questions anytime in the chat, and we'll try to pick up your questions first. If you mark it, this is as a trainee's question. Okay, thank you very much. So we're going to take a break and resume again, I guess, at 10:40. Thank you. Thank you very much.
Hi, I'm Qinghua Kiang from Rice University, from Physics and Astronomy Department. And I'll keep today's the session going. I'm trying to figure out how to use this, so hopefully things will go smoothly. First, I'll introduce the first speaker. The first speaker is Hong Wang from North Carolina State University. She's our keynote speaker here, and she's going to talk about coenzyme SA1 and SA2 are RNA binding proteins that localize to RNA containing regions on DNA. And I thank the speaker.
First, I would like to thank the organizers for giving me this opportunity to share our work. And thank you for joining me this morning. Today, I'm going to talk about two systems. The first system is the telomere system. This is the system that got us interested in cohesion proteins. Telomeres are the protein and DNA structures at the linear chromosome ends. In humans, the telomeric DNA contains 2 to 20 kbs of double-strand DNA and a 3' single-stranded overhang. There is a dedicated group of protein called sheltering protein that are recruited to the telomere region. These proteins include TF1, TF2, RAP1, TIM2, TPP1, and POD1. So the main function of the telomere proteins are to regulate the DNA damage signaling, including ATM and ATR signaling, as well regulating DNA repair pathways at telomeres. So we can think the telomeres at the aglet at the end of the linear chromosome, protecting them from degradation and protecting them from inducing DNA damage response. The second system I'm going to talk about is the cohesion system. So for the cohesion complex, it consists of 4 proteins, SMC1, SMC3, RED21, and the 4th subunit can be either SA1, also called STAC1, or SA2, also called STAC2. So the cohesion complex entraps the DNA inside the ring and promote sister chromatin cohesion, restart of replication forks, DNA double-strand break repair, 3D chromatin organization. However, previously, it was thought that cohesin SA1 and SA2 only function as accessory proteins, promoting protein and protein interactions between cohesion proteins. However, recently, there are more studies pointing to key roles of cohesin SA1 and SA2. For example, one study identified STAC2 or SA2 as one of the 12 genes mutated in 4 or more types of cancer. So this new data tell us that SA1 and SA2 are very important for promoting genome stability. However, on the other hand, we really don't know much about their biophysical properties. Let me go back to the telomere system. When I started at NCC in 2012, the first set of telomere protein I look at were TF1 and TF2. These 2 proteins are only short-chain proteins that directly bind to telomeric double-strand DNA. They are twin proteins. They share the same TFH domain that allow them to dimerize, and also a MIP domain allows them to bind to telomeric DNA. On the other hand, they have very different functions at telomeres. TF2 is crucial in capping and protecting chromosome ends. As shown in these images, deletion of TF2 leads to massive fusion of linear chromosomes right at the telomeres. TF2 has function outside telomeres. It has minor role in telomere length regulation. On the other hand, TF1 plays an important role in regulating telomere length, and it is a dedicated telomere protein. When I started as a biophysicist, one question intrigued me was how do TF1 and TF2 recognize repetitive sequences? To address that question, we use an assay called DNA title assay. It is very similar to the curtain assay, but it's a low-tech version. It's a versatile version. We coat the silica beads with polyol lysine, and when we flow the DNA into the flow cell, some of the DNA will attach to the beads. And when you stretch, some of the stretched DNA can touch the second beads, then a DNA title is formed. We track the protein on the DNA title using quantum dot label proteins. And this platform was developed by NewCat in collaboration with Ben Van Houten when I was a postdoc in Ben's lab. To study TF1 and TF2, Jiang Guo, the first postdoc working in my lab, developed a unique DNA substrate for studying TF1 and TF2. He started with a linear DNA containing 270 TTA-TTT repeats, and then ligate them into longer pieces so that we can string the DNA between beads. So on the DNA title, a unique feature is this. So we will have alternating non-telomere and telomeric regions, and the telomeric region is spaced at defined distance. We conjugate TF1 and TF2, which are histacked to strobotafid encoded quantum dots using a unique biotin NTA compound. So again, this is our ligated T270 DNA substrates. And these are examples of the chymograph for TF1 and TF2. You can see that TF1 is a dedicated telomere protein. It diffuses in a short range consistent with the range of the telomeric region. On the other hand, the TF2 is more venturous. So it has short range diffusion within the telomeric region, but also it wanders into non-telomere regions. This is consistent with TF1 is a dedicated telomere protein, and TF2 has some function outside telomeres. This system works pretty well for TF1 and TF2. Next, I want to switch gear and introduce more cohesion SA1 and SA2, which is the main focus of this talk. This is the domain structure of SA1 and SA2. SA1 and SA2 are twin proteins. They share the same SA domain. They differ on their N termini. SA1 has an AT hook domain. Susan Smith's lab a couple years ago showed that SA1 functions as telomeres and SA2 functions as centromeres. These are her data. Knocking down of SA1 using siRNA leads to defective telomere cohesion, but the centromere cohesion is functional. On the other hand, knocking down of SA2 leads to defective centromere cohesion, but the telomere cohesion is functional. They conclude that cohesion SA1 function at the telomeres and cohesion SA2 function at centromeres. Furthermore, they found sister telomere cohesion is very unique. Largely, it doesn't rely on the cohesion ring. On the other hand, it relies on 3 proteins together. 2 proteins are the short-term protein I talk about, TF1, TIM2. The third protein is cohesin SA1. Their results raise very important questions. How do SA1 and TF1 promote telomere cohesion, which is DNA-DNA pairing without a ring? To answer that question, we are interested to know whether or not SA1 directly bind to the telomeric DNA, or SA1 and SA2 together promote DNA-DNA pairing. In a broader sense, we know cohesin is important for DNA replication fork starts. We know at DNA replication fork, they are single-stranded regions. Also, cohesin is important for DNA double-strand break repair. It has been shown that cohesin prevents the end joining of distal ends. We were wondering whether or not SA1 and SA2 bind to these intermediate structures during DNA replication and repair so that it recruits cohesin to these important structures. Also, do SA1 and SA2 have different DNA binding properties? To address these questions, again, we use the DNA title assay. We use the same ligated T270 DNA substrates. We conjugate SA1, this time flag tagged, to antibody-coated quantum dot using the antibody sandwich methods. We found there are 2 populations for SA1, static and mobile ones. For the mobile one, we saw this classic free 1D diffusion and also this interesting behavior of switching between fast and slow diffusion. You can see the switching behavior more clearly by looking the analysis of diffusion constant over sliding window of 2 seconds. There are 2 distinct populations, slow diffusion and fast diffusion. And this slow diffusion behavior depends on the telomeric substrate. On the telomeric substrate, there is significant higher population of molecules spend more time in the slow diffusion mode compared to other substrates, genomic and centromere DNA substrates. I will show you one video so you can see in real time the pausing and switching behavior. So this pausing and switching to fast diffusion. And we have evidence to support that the pausing events is mediated by the 80 hook domain on SA1. This is the NMR structure of the 80 hook domain from HMG protein. We believe that the 80 hook domain on SA1 sitting at the minor group on DNA cause the pausing events on DNA at the telomeric regions. In comparison, if we mix SA1 and TF1 together, we see some distinct activities. Here, with TF1 alone, it promotes DNA-DNA pairing, short stretches on the order of 60 nanometer. SA1 is a larger protein. It binds mostly individually along the DNA. I should say this is the same linear DNA, the short linear fragment 5.4 kb base pair. And strikingly, when we mix these 2 proteins together, we see much longer DNA pairing tracks compared to TF1 alone. In comparison, SA2 is DNA sequence independent. My former graduate student, President countryman, look at SA2 on different DNA substrates, centromeric, telomeric, and lambda DNA. On all these substrates, SA2 shows similar diffusion constant. This shows that SA2 doesn't care about the sequence. It is a sequence-independent DNA binding protein. This is consistent with their structure. SA2 doesn't have the 80 hook domain that enables SA1 to pause at telomeric regions. In addition, we made DNA substrate containing special features. Here, we made DNA substrate containing 37 nucleotide single-strand gap, 23% from the DNA ends. We look at the binding position of SA1 and SA2 on this substrate. SA2 mostly positioned at the DNA ends and at the single-strand DNA gap. On the other hand, the distribution of SA1 on this substrate is random, and SA2 distribution on the substrate without the gap is random. We can also see in real time the binding of SA2 on the gap region. So here we made the DNA title with single-stranded DNA gap at defined spacing 2 kb. At high concentration, we saw SA2 load onto the DNA title with defined spacing, consistent with the spacing between single-strand DNA gap. At low concentration of SA2, we see again this switching behavior between fast diffusion and stable binding. In addition, we made a more complicated structure mimicking DNA repair and replication intermediates, a fork and replication fork positioned at 23% from the DNA ends. We analyze the position of SA2 along these substrates. SA2 mostly positioned at the flap, fork, and replication fork structures. Furthermore, to understand where exactly SA2 bonds to on the replication fork, we made this longer DNA substrates using a plasmid obtained from Jack Griffiths. You can see here most SA2 bind right at the junction at the replication fork. So these images tell us SA2 can bind specifically to DNA replication and repair intermediates. Here, I want to summarize the first part of the talk. At telomeres, we can think TF1 as the local resident. It diffuses by itself within the telomere region, and SA1 and SA2, we can think them as the housekeepers along the genome. They are the travelers on the DNA. We saw the searching mode, fast diffusion, and the recognition mode. For SA1, the recognition happens at the telemetry sequences. And for SA2, the recognition happens at the single-stranded gaps. We also saw SA2 bind to flap and replication fork structures. One piece of data I didn't have time to go into detail is that when we do color label both TF1 and SA1, we saw that TF1 tethers SA1 at the telomere regions. So TF1 as a resident at telomeres recruits the traveler SA1 to the telomeres. TF1 and SA1 together promotes DNA-DNA pairing. We were also looking at the function of TIN2. We have a paper in revision right now showing actually TIN2 can facilitate TF1-mediated trans and cis interactions on long telomeric DNA. So this has been already posted on bioRxiv. Next, I would like to switch gear to share with you how we discover SA1 and SA2 are RNA binding proteins. When I presented our data on SA1 and SA2 DNA binding at the Gordon Conference in Hong Kong in 2016, Alex Bishop presented at the same meeting on immune sarcoma. What they found was in immune cells, there are higher level of R loops compared to control cell lines. R loops are 3-stranded nucleic acid structures comprising RNA DNA hybrids and non-template single-stranded DNA. So we connected at the meeting and also connected some dots. I learned that EWSL1, the most commonly mutated protein in immune sarcoma, is an RNA binding protein. And the second most common mutations in immune sarcoma is in the cohesin SA2 gene, happening in about 15% of immune sarcoma patients. At that time, we were wondering whether or not cohesin SA1 and SA2 bind to RNA and R loop structures. And this can explain the function of SA2 in sarcoma. To address that question, again, with the help of Jintao, we purify all three proteins. And we first made the model R loop substrates. In substrates, we labeled the RNA component with fluorophore. And this native gel showed that indeed we form three stranded structures. And we validate the substrate by using S9.6 antibody. The antibody bind to the model R loop, but not the double-strand DNA. Our answer gel showed that SA1 and SA2 bind to the model R loop. Jintao's group shows using the fluorescence anisotropy, SA1 and SA2 bind to R loop at low nanomolar concentrations. In addition, they did a more extensive study. They show that SA1 and SA2 binds to single-stranded RNA in a length-dependent manner. They also tested various RNA substrates on SA1 and SA2. SA1 and SA2 bind to double-stranded RNA with overhang, RNA DNA hybrids with overhang, and RNA DNA hybrids. Combining our previous study of SA1 and SA2 to DNA, a striking feature came out. For the single-stranded RNA and DNA with the same length, SA1 has 50-fold higher affinity, and SA2 has 35 higher fold affinity for the single-stranded RNA compared to single-strand DNA. So before I told you that SA1 and SA2 are DNA binding proteins, actually they are RNA binding proteins. The data I show you on shorter RNA. The question is whether or not cohesive SA1 and SA2 combine to longer RNAs. This longer RNA can form a secondary structure and prevent protein binding. To address that question, Ming Liu, a graduate student in our lab, made longer RNA transcript using the mega transcript kit. This is the image of the transcript after purification. These are the images after incubating SA1 and SA2 with the RNA transcript. You can see that with SA1 and SA2, there are features with taller heights and larger volume, indicating that SA1 and SA2 can bind to longer RNA transcript. Next, to study the RNA binding of SA1 and SA2 on longer DNA substrate, we made this unique substrate following the protocol provided by Chetin's group at UC Davis. We transcribe first on this substrate containing a G3 promoter. Then we trim the free RNA using RNase A. And after digestion, we have two pieces. One piece has R loop over the two regions containing consecutive Cs, and another control DNA. AFM images show that there are unique features, the spur-shaped, loop-shaped, and blob-shaped structures forming on the longer substrates. In addition, we validate the substrate using S9.6 antibody. Here, we incubate the linear R loop substrate with S9.6 antibody. Upon incubation of the antibody with the R loop, we saw taller features compared to R loop alone. And both of the R loop feature and the feature upon incubation with the antibody positioned at 40% from the DNA end, consistent with the design of the substrate. On the other hand, the distribution of the antibody on the double-strand DNA is random. So these results validate the formation of R loops on our substrate. Then we can look at the R loop binding by SA1 and SA2. Here, we look at the protein complexes, which display taller feature compared to R loop alone. We look at their position along the DNA from one DNA ends. Their positions are consistent with the position of the R loops. In comparison, these three proteins position randomly along the double-strand DNA substrates. So these AFM image demonstrate that SA1 and SA2 can directly bind to regions containing R loops on the double-stranded DNA substrates. In addition, we look at the dynamics of SA1 and SA2 binding to R loops. Here again, we use the DNA title assay. We conjugate the Histag protein to streptavidin-coated quantum dots. We ligate the DNA first, then generate R loops on the substrates. The spacing between the R loops is about 1.4 micrometer. We expect to have about 8 R loops per 10 micrometer of DNA titles. These are the examples of SA1 and SA2 on the R loops DNA titles versus the control DNA. Two features stood out from these comparisons. First, you can see that these proteins are socially distanced on the DNA title. If we look at the spacing between the proteins for R loops, they are positioned on average with distance about 1.5 micrometer, which is consistent with the spacing between R loops. So on the R loop substrates, SA1 and SA2 recognize these R loops. On the other hand, for the control substrates, the density of the protein are lower and they are randomly distributed along the DNA. The next question is, since we observed the RNA binding by SA1 and SA2 using single molecule techniques, do they bind to R loops in vivo inside the cells? To address that question, Henry Miller, a graduate student in Alex's lab, did some extensive analysis. He put together all the available ChIP-seq data for SA1, SA2, SMC1, SMC3, and cohesin interacting protein CTCF. He put all the ChIP-seq data to get binding position for these proteins. And also he put out current data sets for the location of R loops identified by ChIP-seq. From his analysis, you can see that for all 4 cell lines, about 20 to 50% of the drip peaks overlaps with SA1 and SA2 peaks. So indeed, these analysis suggests that SA1 and SA2 can bind to R loops inside the cells. Furthermore, he addressed the question whether or not SA1 and SA2 localized with other cohesin subunit, SA1 and SA2 together at the R loop. This is his analysis showing that R loop localized SA1 and SA2, which is labeled as SA2OL drip. It overlapped with the position of SMC1 and SMC3. This data again show that SA1 and SA2 are in complex with the whole cohesin complex when they bind to the R loop region. In addition, he analyzed the distance to the transcription start sites for different data set for SA1 and SA2 and SA1 plus SA2 and SA1 plus SA2 at the R loop site and SA1 and SA2 without R loops. This analysis showed that in all 4 cell lines, the SA1 and SA2 localized at the R loops are closer to the transcription start sites. Here I want to summarize what I have shown. both cohesin SA1 and SA2 are DNA and RNA binding proteins. They bind to single-strand DNA and double-strand DNA. They have higher affinity for single-strand DNA. SA1 specifically recognizes telomeric sequences, and SA2 recognizes single-stranded DNA gap, flap, and DNA replication fork structures. One caveat in this statement is that we believe that SA1 also recognizes single-stranded DNA gaps, DNA flap, and replication fork, but due to the competition binding at the AT-rich sites, if both SA1 and SA2 are present, SA2 will be the go-to protein that recognize these spectral structures. In addition, we found that SA1 and SA2 are RNA binding proteins. They display significant higher affinity for RNA compared to DNA. They bind to various RNA substrates, including double-strand RNA, RNA-DNA hybrids, and R-loops. Some future directions, as I shown you, we understand that TF1 is a local resident at the telomere region. It can recruit other genome maintenance proteins, such as SA1. And right now we are trying to understand the role of RNA binding by SA1 and SA2 at telomeres. We made telomeric R-loops. Our preliminary data show that SA2 indeed bind to telomeric R-loops. Also, we are doing some live cell imaging to study the recruitment of SA1 and SA2 at telomeres under DNA damaging conditions. Here, I show you the telomere foci marked by M-cherry TF1 and the pattern of EGFT SA2. So finally, I would like to thank people in the lab who have done the work, very talented Haipan, Pamina Kerr, and Ning Liu, and the undergraduate student who contribute to the R-loop study, and people in Patty's lab who provides us with protein. Susan provided us with TIN2 protein. Jacob provides us with quantum dots. Jack gave me the telomere substrates. Neil helped me with the DNA tightrope assay. Especially, I want to thank Jean for continuous support in the past few years to provide us with cohesin proteins. It is critical for our project. And Alex for the R-loop project, and Peggy for providing us the substrates, allow us to make the gap DNA substrate, and our funding agency. And thank you for joining me today, and thank you for your attention. Any questions?
Thank you. So now we're open for questions. Please type your question in Bella. Let me switch to the view. So if you you need to open the
yes,
Bella,
yes,
right?
Yes, yes. So I will answer by sequence. So 80 hook mutants of SA1 variable, and are there telomere-related phenotype in cells? Yes, Susan Smith's lab showed that 80 hook mutant is defective in telomere cohesion. And this question about whether or not cohesin is looping DNA at R loops, we pretty much believe so. There's recent study showing that SA2 is recruited to super enhancers. We think that cohesin SA1 and SA2 are the sequence and structure guides for the cohesin complex. They can anchor the cohesin complex at regions containing RNA and loops the DNA. I think this is something we will test further using single molecule studies. Do you have model for how the recognition works for gap forks, and is this structure specific? This is a great question. It is a very complicated a protein, very versatile protein. Currently, Jin and our lab are making mutant proteins of SA2 and test the DNA and RNA binding. Hopefully we can identify the DNA and the RNA binding domains on SA1 and SA2, and maybe we can find a domain that's specific for DNA and specific for single-strand DNA versus specific for double-strand DNA. So it's still ongoing. The terra ink RNA is transcribed from subtelomere region, has been shown by, to form alloway telomere. Would be interesting to see. Yes. If SA1 and SA2 recruit telomeres via, yes. So this is what we are going. We just made the short DNA substrate containing the telomere allo. And from AFM imaging, it looked like SA1 and SA2 is recruited to the telomeric allo. We'll also have data showing that the TF2 also recruits telomeric RNA, terra RNA to the telomere region. Yeah, definitely this is a very interesting direction. In general, the emerging theme is that RNA forms on DNA serving as a scaffold to recruit other proteins and to introduce new functions along the DNA. So our study identifying SA1 and SA2 as RNA binding protein fits this scheme. So with only DNA, it might be boring and limited, right? So now you have a second player with RNA, and you can recruit other proteins under specific conditions. Yeah. Thank you for all the comments.
We have any more questions? Please type in umbrella. We have just a little bit time for one more question. I have one quick question. You talk about the fast and slow diffusion of, I think SA2. What's the significance of that? Is there any biological functions that requires them to have two different diffusion?
That's a good question. So these proteins are housekeeping genes. They will diffuse along the DNA by themselves, right? But then when they encounter specific sequences, for example, RNAs, they will stop and then recruit other cohesin components. So they are kind of the traveling guides for the cohesin, right? So they tell the whole cohesin complex where to stop, where to function. In the telomere, which is another example, without TF1, SA1 will pause at the telomere just for a short amount of time, right? When it encounters TF1 will say, oh, stop here, we have some function to do. And these two proteins will stop at telomere and function there. Okay. So in general, I think the pausing and stopping events really tell us where this protein function.
Okay. No more questions. Let's thank the speaker. Thank you. We'll start the next speaker.
Yes, let me stop the share.
Okay. Okay. Great. And our second speaker is Anna Karen Gustafsson from Rice University. She just joined the Department of Chemistry at Rice. She will talk about 3D single molecule super resolution light. 3D single molecule super resolution light sheet imaging through our mammalian cells.
Thank you so much for that introduction. That title is a handful, I know. So yeah, once again, thank you for that introduction, and thanks a lot to the organizers for organizing this event and for inviting me to present our work here today. So today I would like to share with you some of our work on the 3D single molecule super resolution imaging using light sheet illumination to image entire mammalian cells. And just like you heard, I recently came to Rice. So the work that I'll show here today was done over at Stanford University together with W.E. Murner. But here at Rice, we are continuing to develop these techniques and using them for various applications in biophysics and biomedicine. So if you find these techniques interesting, please feel free to reach out to me, and I'm always happy to discuss potential collaborations. So let's get started. So conventional cell imaging is limited in resolution by the diffraction of light. So this means that we cannot resolve features that are closer together than about 200 nanometers. So we can easily resolve macroscopic objects, such as a strand of hair, whole cells like mammalian cells or bacterial cells, and some subcellular features like mitochondria. But we are unable to resolve any nanoscale features within these cells. Luckily, there are ways to circumvent the diffraction limit. And in our work, we rely on single molecule imaging to do this. And here we rely on two key ideas to achieve super resolution. And the first is super localization of an emitter. So this technique has in principle been done throughout several of the talks today, but I'll go through some of the basics. So if we know that we're imaging a single fluorophore or a single emitter on our camera, we will see an intensity distribution looking something like this, describing the point spread function, or the PSF, of our microscope. If we know that we're imaging a single emitter, we can estimate this intensity distribution with a model function, and from this estimate the center position of this distribution with a precision that is much finer than the width of the distribution itself. So this is what we call super localization. And the precision that we can achieve scales roughly as 1 over the square root of the number of photons that we detect. So we want to detect as many photons as we possibly can from the fluorophores. But it also depends on the background fluorescence from the rest of the sample. And this is an important factor that I'll come back to later on. The second key idea is active control of the emitter density. So if we have a densely labeled structure and we allow all of the fluorophores to emit at once, all we'll see is a diffraction limited blur. But if we can actively control the density of emitters that is allowed to emit in a single camera frame and keeping that density low, then we can apply key idea number 1 and super localize each of these emitters. In the next camera frame, we can allow a different sparse subset of molecules to emit, super localize those, and over time build up a super resolved reconstruction of this underlying structure. So these two key ideas are used in multiple different techniques that you may be familiar with, such as PALM, STORM, FPALM, and what we collectively like to call them SMAC, or single molecule active control microscopy. So the goal with our work is to extend these techniques for 3D single molecule tracking and 3D super resolution imaging throughout entire mammalian cells. And there are a few challenges to this. The first is, of course, that cells are three-dimensional. So we need a way to detect the position of single molecules on the nanoscale in 3D. The second challenge is the sheer thickness of these mammalian cells in contrast to more conventional super resolution targets like bacterial cells. And this thickness will create a lot of background haze in our images that will degrade our localization precision and, in some cases, prevent imaging altogether. So our solution is to combine point spread function engineering for detection of single molecules in 3D with light sheet illumination for optical sectioning of these thick cells. So I'll walk you through these techniques in some detail, starting with PSF engineering. So if we're once again imaging a single molecule on our camera, we'll see the standard point spread function of the microscope looking something like this. And if the molecule is moving up and down, we will see how this comes into focus and then blurs away from focus. However, this standard PSF changes slowly near the focal plane. It has a limited axial range before it blurs out of about 500 nanometers, and it's symmetric about the focal plane. So this makes it less than optimum for imaging of single molecules in 3D. So what we do is to introduce a phase pattern in the Fourier plane of the microscope. And this phase pattern adjusts the phase of the light that is emitted from the single molecule to encode the axial position or the C position of the emitter directly in the shape of the PSF on the camera. So in the example you see here is the double helix PSF. And instead of consisting of a single lobe that blurs away from focus, we here have two lobes that revolve around each other. The midpoint between the two lobes encode the XY, or the lateral position, and the angle between the two lobes encode the C position. So in contrast to the standard PSF, the double helix changes rapidly near the focal plane. It has a variable axial range of between 2 to up to 10 microns, and it's non-symmetric about the focal plane. So this makes it very useful for imaging of single molecules in 3D, and it's been used extensively for imaging in bacterial cells, and as I'll show, also more recently in mammalian cells. Another set of PSFs were recently developed to be maximally informative over user-specified axial range. And these are called the tetrapod PSFs. And they consist of a slightly more complex pattern than that of the double helix, but it roughly traces out the shape of a tetrapod when seen in 3D. And just like the double helix, the tetrapods also change rapidly, can be made with a variable axial range, and are non-symmetric about the focal plane. And these have been demonstrated with axial ranges of up to 20 micrometers, which is a very long axial range. And this makes them useful for tracking of bright and sparse emitters, like tracking of a fiducial bead for drift correction, or tracking of chromosomal loci. And more recently, they've also been demonstrated for single molecule super resolution imaging. Okay, so now we have tools to detect single molecules and fiducial beads in 3 dimensions with very high precision. So next, let's take a look at light sheet illumination for optical sectioning of these fixed cells. So light sheet illumination was originally developed for imaging of larger scale samples, such as embryos. And here the light sheet is typically formed using very simple optics, such as a cylindrical lens that focuses light in 1 dimension onto a plane or a sheet of light. And this sheet is then introduced from the side to optically section the sample. But if you try to use this pretty straightforward method for imaging of single molecules in single cells, you run into some issues. And the reason is that for single molecule imaging, you want to detect as many photons as you possibly can to improve your localization precision. And this means using objective lenses with high numerical aperture, which then also will have a short working distance. So you will need to place your sample very close to the objective itself. And for mammalian cell imaging, that typically means having the cells directly on the cover slip. But when you then try to introduce a light sheet from the side to section the cell over here, you will introduce aberrations in the light sheet in this interface between the sample chamber wall and the bottom cover slip. And these aberrations will degrade the light sheet and reduce its optical sectioning capability. So because of this, there's been several different designs developed to try to circumvent this issue, but they all have drawbacks in certain situations. So in many cases, they consist of complex designs with many custom-made parts that are hard and expensive to implement in many laboratories. In many cases, they have difficult sample mounting, once again with custom-made parts. And in some cases, they have increased risk of sample contamination due to dipping of objectives into the sample chamber itself. And for single molecule imaging in cells, this problem is twofold, in that you both risk introducing biological contaminants, which of course can be detrimental for cell imaging, but you also risk introducing single fluorophore contaminants. So keeping in mind, we can detect the light from single molecules, so even single fluorophore contaminants can degrade our images. And despite these drawbacks, some of these designs still suffer from a gap very close to the cover slip that is inaccessible to the light sheet, and thus prevents imaging of entire adherent mammalian cells. So our solution was to simply tilt the light sheet. This allows us to still form the light sheet using the very simple optics, such as a cylindrical lens, but still introduce the light sheet away from this aberrating interface here. We can use conventional cover slips for culturing and imaging, a conventional high numerical aperture objective, and a conventional microscope body. We don't need to dip any objectives into the sample chamber, reducing the risk of sample contamination. And despite the simplicity, we can still access entire adherent cells all the way down to the bottom. You might note here that there will be an angle difference between the image plane, where we detect the single molecules, and the light sheet plane, where we excite them. But this is not an issue when we combine this tilted light sheet design with our 3D PSFs. Because here we will be able to detect all the single molecules in the entire axial range that is excited by the light sheet. And keep in mind that the precise position of the single molecules will be determined by the shape of the PSF on the camera, rather than by the precise position or thickness of the light sheet. So here you can see a schematic of our experimental design that we have termed tilted. So here we simply form the light sheet using a cylindrical lens that focuses light in one dimension onto a motorized mirror. This motorized mirror plane is then relayed by two lenses in 4F configuration onto the back aperture of a long working distance illumination objective. This objective lens then focuses the light sheet that is reflected by a prism at an angle or a tilt into the sample here. By tilting the motorized mirror, we can translate the light sheet both up and down, so in the Z direction, and in the image plane. And here to the bottom left, you can see a photo of the light sheet in action, where you have the illumination objective with the light sheet being focused down and being reflected by total internal reflection at the back of this prism into the sample chamber and being focused down right at the bottom over here. So we've shown that by using this simple light sheet design, we can improve our signal to background ratio more than by a factor of 5 compared to conventional epi-illumination. So this is shown here for the nuclear lamina, which is a protein meshwork that lines the inside of the nuclear envelope. And this improvement drastically improves our localization precision for single molecules, as I'll show later on. The next step is to detect the light that is emitted from these excited molecules. And we are doing this using a conventional high numerical aperture detection objective, a conventional microscope body, but instead of placing our camera right here at the port of the microscope in the first image plane, we are relaying this image plane by lenses in 4F configuration onto a new image plane over here where we place our camera. And in this case, we are splitting light into two different color channels. So far red light goes this way and greener light goes this way. And by doing this two-channel setup, we can independently introduce different face masks in the different channels. So we can introduce a double helix face mask for precise detection of single molecules in the far red channel. And for example, a very long axial range tetrapod PSF or face mask in this channel for detection of fiducial beads for drift correction over a very long axial range. So next we set out to benchmark this imaging and we started with imaging of mitochondria. I'm not sure if you can see the top right here, but to keep you on track, I've included a schematic here where we have outlined the labeled structures in red, so in this case, mitochondria. And this will keep you on track also when I show some other structures later on. So to the left here, you can see a 2D diffraction limited image of mitochondria, but from this image, we cannot really see any nanoscale features. So we then turn to the single molecule regime. And here you can see individual molecules blinking on and off, where each blink consists of 2 lobes, which is the footprint of the double helix PSF. And if you look very closely, you can even see that the angle between the lobes for the different blinks differ. So this shows that there is some 3D structure in the sample. By localizing these molecules, we can then create a 3D super resolved reconstruction of this underlying structure. And in these plots, the color encodes for the C position, so the axial position of the molecules. And here we drastically improve our resolution compared to diffraction limited imaging. So we can even look at individual mitochondria. So here you can see an XY, an XC, and a YC projection, which clearly shows the hollow structure of the mitochondrial outer membrane that was labeled in this case. We can also look in more detail at this intricate network that is formed by these mitochondria. And we can even look at this from different angles because we have the full 3D information. So in this sample, we made a direct comparison between using our tilted light sheet design and using conventional epi-illumination for illumination. And we saw a dramatic improvement in our localization precision, both in XY and in C, as you can see here, demonstrating the strength of this platform. So next we set out to image an even more challenging sample. And here is the entire nuclear lamina of a cell. So the nuclear lamina is this protein meshwork that lines the inside of the nuclear envelope that I showed before. And we are interested in understanding the structure better because some of these proteins are involved in several different diseases called laminopathies that we want to understand better on the molecular level. So here we show that we can image this entire structure throughout the entire cell with good localization precision throughout the entire cell and with a consistent lamina thickness detected of about 100 nanometers throughout the entire nucleus. We can look at different parts of this structure. So here I show 100 nanometer thick C slices through this reconstruction. So here you clearly can see these undulations and folds at the bottom part of the nucleus in the blue region coming up right here. And we can also take a closer look at this thin rim at the nuclear periphery, this roughly 100 nanometer thick rim. We can also take a closer look at some structures of interest. So here instead we have sliced the nucleus in the Y direction. And here we have isolated an intranuclear lamina channel that spanned all the way across this nucleus. So these are the types of structures that we are interested in studying further and that we can easily resolve using our platform. So the next project I would like to show you is a collaboration with the Bertozzi lab at Stanford. And here we were interested in looking at the nanoscale distribution of sugars in the glycocalyx and how it's affected during cancer progression. And glycocalyx literally means sugar coat, and it's a good description of what it really is. So it's a sugary coat that lines the outside of most of the cells in our bodies. It's the first thing that comes in contact with the extracellular environment, and it's important for proper development, but also for metastasis in cancer progression. So we first set out to image these cells using 2D diffraction limited imaging. And just like before, we could see the overall structures, but not really resolve any nanoscale features. So we then turned to 2D super resolution imaging. And here we could clearly resolve these, what we saw from the 2D diffraction limited imaging, these apparent membrane protrusions or tubules. And from the super resolution imaging, we can resolve these as hollow. So this means that the glycocalyx is really coating these protrusions that are a characteristic of cancer cells. And this gives us a way to extract the thickness of the glycocalyx along the length of these tubules. However, from 2D imaging, we cannot really characterize the thickness and the distribution of sugars throughout the entire length of the tubules in the cases where we are losing the tubules out of the plane or out of the field of view here for 2D. And we also cannot see how this distribution might be changed or varies across the entire surface of the cell. So to do this, we turn to 3D super resolution imaging. So this is the reconstruction from this sample that you saw over here to the left. And here we once again could clearly resolve these tubules as hollow, and we could quantify the thickness of the glycocalyx along the entire length of the tubules, even in the cases like here when the tubule was directed vertically. We could also distinguish some ambiguous cases like the apparent blobs that we saw in the 2D reconstructions. Are they actually blobs or are they tubules directed vertically? And from these images, we could see that in all cases, these were tubules that were directed out of the plane. And here's another example of an ambiguous case where two tubules appear to merge at the top. But since we can look at this from different angles, we could actually determine that these are simply wrapping around each other. So from these images, we could quantify both the thickness or the width of the tubules along their entire lengths and found it to be quite consistent. And we could map the thickness of the glycocalyx for various conditions. And particularly, we found that the thickness of the glycocalyx was increased during two different hallmarks of advanced human cancer. So during oncogenic KRAS activation and epithelial to mesenchymal transition. So by doing this type of imaging, we could directly measure this increase in the glycocalyx thickness. So we're hoping that this discovery will help guide future development of drug treatment strategies that can help in cancer treatment where the glycocalyx is affected. So the last project I'll share with you today is a collaboration with the Jackson lab at Stanford. And here we were interested in understanding the nanoscale distribution of proteins in the inversing compartment in the primary cilium. So the primary cilium is a sensory organelle that protrudes from the cell membrane in many of the cells in our bodies. And the inversing compartment is a subcompartment of this primary cilium to which 4 proteins are known to localize: inversin, ANC6, NAC8, and NPHP3. The function of the inversing compartment remains unknown, but it's been shown that mutations in these proteins cause complete reversal of symmetry of the organs in our bodies, as well as cystic kidneys. So they obviously have an important function. So we then set out to investigate what is the 3D nanoscale structure and organization of these proteins in the inversing compartment. But as you can see here, the entire width of the primary cilium is roughly on the order of the diffraction limit. So we clearly needed to turn to super resolution imaging to answer this question. So here we started out imaging inversin, one of these inversing compartment proteins, and started out with diffraction limited imaging. And as expected, we couldn't really resolve any structure at all. So we then turned to 3D super resolution imaging. And here, strikingly, we found a previously unknown structure that is formed by this protein. So instead of being homogeneously distributed throughout the inversing compartment, this protein forms these fibril-like structures or fibrilloids that span along the ciliary axis. So we could investigate these structures from different angles, and we could also look at cross-sections through different parts of the inversing compartment and quantify, for example, the number of fibrils or fibril-like structures along the length of the inversing compartment. So this finding was quite intriguing. So by studying this in some more detail, we decided to set out to image also the other proteins. And here you can see both inversin, ANC6, and NPHP3. And strikingly, they all form this fibril-like structures. So we quantify the length of the inversing compartment, the length of the individual fibrils, and the number of fibrils per primary cilium, and found it to be quite diverse in all cases. We then turned to 2-color super resolution imaging, but also imaging microtubules in the axone, which is this center part of the primary cilium. And here we found that these fibrils are wrapping around the axone. So this project raised a lot of new questions, such as how are these fibrils formed? Why are they formed? And how do they affect the function of the inversing compartment? So these are all questions that we would like to address in the future. So with that, I would like to wrap up and say that 3D single molecule super resolution imaging throughout entire mammalian cells can reveal both nanoscale features and molecular mechanisms in healthy cells and in disease progression. So I've shown you some of the techniques that we're using and showcased examples of imaging in mitochondria, the nuclear lamina, the glycocalyx and how it's affected during cancer progression, and our discovery of a new type of structure in the primary cilium. So these techniques can not only be used for super resolution imaging, but also for live cell single molecule tracking in 3D. So this is something that we're working on right now. We are also working on extending the capacity of this platform for very precise multicolor imaging. And this will allow us to do co-localization studies and looking at molecular interactions. And we're also interested in various biophysical and biomedical applications. So as I said in the beginning, if you're interested in these techniques, I would be very happy to talk and discuss potential collaborations. I would also like to put it out there that we are looking for enthusiastic postdocs to join our team. So if you are interested in, or if you know of a suitable postdoc candidate, then please reach out to me. So with that, I would like to thank the entire group over at Stanford that were involved in the work that I showed today, our fantastic collaborators, both at Stanford and elsewhere around the world, and our funding agencies for making all of this work possible. And of course, all of you for your attention. Thank you.
Thank you. So now we're open for questions. Let's look at Brella. Please type in your questions in Brella. Let me see. The first.
Why don't I get rolling just asking you, have you used this technology to image things like phase separations, particularly in the nucleus, or which are membranous organelles, and do you think it could work for that?
Yes, so in principle, what we can label, well, we can image. So the main limitation is really the labeling. How would you label the different structures that you're interested in? We have, well, the inversing compartment is a membraneless organelle in a sense, but here we were targeting the specific proteins. And the same goes for some bacterial imaging, where we're looking at POPC and other membraneless self-organization parts of the cell. I don't know if that addresses your question.
Okay. The next question is, do you need to fix cells to image the nucleus? Is phototoxicity a problem in live cells?
So the question to the two questions really, it depends, and yes. The images I've shown today is on fixed cells. One avenue that we're pursuing right now is for live cell super resolution imaging. Tracking we can easily do in live cells, whereas super resolution imaging typically requires high laser powers, toxic buffers to make the molecules blink. But recently there's been a lot of development for live cell compatible fluorophores that are blinking also at lower laser powers and non-toxic conditions. So that's something that we are working hard on right now to address. So I'm hoping to in the future be able to correlate both tracking of dynamics and structural context. But yeah, for the examples I've shown here today, these are fixed.
Okay, next questions. Are there special requirements for the fluorescent probes as in lifetime of photo linking for LS microscopy?
Yeah, so there are no specific requirements specifically for the light sheet part, but definitely for the single molecule super resolution part. So you need fluorophores that can be made with some active control mechanism. It can be that they can be photoactivatable, photoconvertible, or photoswitchable. So it can be either fluorescent proteins or synthetic dyes, but we need some way to actively control and have most of them dark. Because if all of them, as I showed in the beginning, emit at once, then we cannot localize the individual ones. So we need this kind of spatial or temporal separation. But yes, when it comes to the time dependence on the blinking, we can to some extent tune that, both with laser power and with the blinking buffers that you use, how fast they blink. So the faster they blink, the faster you can image. So for live cell imaging, that will be an important criterion so that you can actually capture your full reconstruction before the cell moves, so the structures rearrange. But then you typically also get fewer photons per frame which can be limiting when you get too low.
Okay, well, we have a minute, 2 questions. Next one is, are the tubules of glycocalyx entirely made of sugars?
So, sorry, I missed the first part of your question. Are
the tubules of glycocalyx entirely made of sugars?
So the tubules are actually membrane, so they are contiguous with the cell membrane. So the glycocalyx is really just coating the outside of those tubules, but the tubules themselves are not made of glycocalyx, but they are covered by it.
Okay, the last question. Have you taken multiple images to track the motion of the molecules? Is it possible to do so?
Yes, so that's really single molecule tracking, and we can definitely do that. So if you're interested in looking at some specific proteins or some specific molecules that you can tag, you can definitely track them in 3D with this technique. And what we're hoping to do, so yeah, we've done that for chromosomal loci tracking, for example, even with 2-color. What we're working on right now is to get that even more precise, to really be able to look at molecular interactions as well in 3D, how molecules come together on the nanoscale.
Okay, well, now it's perfect timing. Let's thank the speaker. We'll welcome the next speaker. That's the last speaker of this session. Okay, so next speaker is by Vinay Neer from University of Texas Health Science Center in Houston. And he's going to talk about this long-range coupled motions underlie ligand recognition by a chemokine receptor. Okay, please go ahead.
Okay, am I audible?
You're live, go ahead.
Okay. Thank you. I'm Vinay. I'm a graduate student in the lab of Alex Godfrey at McGovern Medical School. And today I'll be talking about this interesting collaboration that we had with the Rajaratnam group at UTMB Galveston, where we were interested in identifying the structural mechanism of the binding of chemokine CXCL8 to its receptor CXCR1. Chemokines are small proteins. They are around 10 kilodaltons in size, and they are secreted by cells of the immune system. And they act as directional guidance cues to other migrating cells of the immune system. They basically bind to chemokine receptors. Chemokine receptors are typical class A GPCRs. They have the traditional GPCR structure of an extracellular N-terminal domain and an intracellular C-terminal domain and the 7 transmembrane helices. Chemokines bind to the chemokine receptors at 2 distinct sites. We have site 1, which is at the N-terminal domain or the extracellular domain. And site 2, which is basically a groove that is defined by the receptor extracellular loops or the transmembrane helices. Site 2 is a very classical GPCR agonist binding site, whereas site 1 is something that is more specific for chemokines. So we know that chemokine binding at site 2 triggers receptor activation. However, the role of site 1 interactions is not known. And within this context, we are interested in CXCL8 or interleukin 8 as it is more commonly known, and the binding of this chemokine to its receptor CXCR1 and CXCR2 and how it affects downstream signaling. So what is interesting is CXCR1 can be activated only by CXCL8, whereas CXCR2, which is the other receptor, is more promiscuous and can be activated by the entire ELR family of chemokines. Now, looking at the similarity between the two receptors, they're fairly similar to each other except at the end domain, whereas where there is a significant difference in the end domain sequences between CXCR1 and CXCR2. And our overarching hypothesis is that the sequence difference between the two receptors is what determines their interactions and the specificity of these interactions. It would be really helpful if we could have more detailed structural information about what is happening at these N-terminal domain. However, the problem is that this N-terminal domain is intrinsically disordered and it is difficult to obtain structural information. So for this project, the aim of our project is to determine the structure of the CXCL8 and the CXCR1 complex and to elucidate the Site1 and Site2 interactions. And towards this, our collaborators at UTMB Galveston, they use the CXCL8 monomer and only the CXCR1 end domain bound to the monomer. And using NMR, they were able to determine the solution structure using distance and angular restraints. And over here, I'm showing an ensemble of the complex that has been obtained from NMR. And what is interesting is that the N-terminal domain, which was previously disordered, becomes structured on binding to CXCL8. They were also able to identify different intermolecular contacts between the chemokine and the receptor. From these contacts and the NMR, they were able to build a structural model using the NMR information and previously available other structural information. And over here, I'm showing the complex that has been generated using the NMR data and homology modeling. So if you look at the Site1 interactions, the Site1 interactions basically take place within this dashed circle that I've shown over here. And the Site1 interactions of the receptor with the chemokine can be broken down into three groups of residues. So first we have the red. So I won't go into the details of which residues they are, but just to give you an overview, the red set of residues over here which are interacting basically have ionic interactions with the receptor. Then we have the green set of residues which form a mix of hydrophobic packing and ionic interactions with the receptor. And then we have the third set of residues, which are the pink residues that you see at the top over here, which are mostly at the C terminus of the chemokine. And again, these have ionic interactions between the receptor and the ligand. And these extensive packing, hydrophobic packing, hydrogen bonding, and salt bridges are what stabilize the complex. So armed with this information and the structural model which we have developed, we wanted to see how the Site2 interactions would take place, knowing the Site1 interactions. And for this, we performed molecular dynamic simulations. And we simulated the entire system for close to 1 microsecond. And we saw that within our simulations, there were large conformational changes that were taking place. There were large conformational changes that were taking place at the N-terminal domain, and also there were Site2 interactions being formed. So to analyze this, so as I said earlier, we have two sets of interactions taking place. We have interactions at Site1, which I've highlighted in red over here, and we have interactions at Site2, which I've highlighted in black. So I calculated the pairwise interactions of the receptor residues at the two sites and the chemokine residues. And there are several things that stand out. So firstly, if you look at the heat map of these pairwise interactions, you can very clearly see the interactions at the C terminal, which are these pink residues over here at Site1. We see the interactions at the green and the red residues, which are these interactions over here at Site1. And we also begin to see interactions at Site2. And these interactions are the blue residues, which you see over here. And these blue residues are basically the ELR motif of the chemokine. The ELR motif, which in this case is present in residues 4, 5, and 6, are very well established to be important in Site2 binding, and mutating any of the ELR residues disrupts Site2 binding. So in our simulations, we see that within the first 50 nanoseconds, we see these interactions beginning to form. And at the end of our simulations, we see several things taking place. So the first thing which we noticed is that a lot of the site-1 interactions were transient. Mainly the interactions at the C-terminal residue are lost, which we see over in the heat map. We see that there is some structural plasticity, which allows residues to interact both at site 1 and site 2. And these residues belong to the red group of residues over here. We also see that even at 800 nanoseconds, where we have firmly established site 2 interactions, there are site 1 interactions which are still dynamic and are changing. And also, we see that the ELR is firmly interacting with the site 2 residues. So the overall model that we have is that the receptor CXCR1 captures CXCL8 by a fly casting mechanism via interactions at site 1, which is over here. And these site 1 interactions allow the ELR residues, which are important for site 2 binding, to scan the conformational space. And after scanning the conformational space, they are able to bind to the site 2. And also these interactions are governed by structural plasticity, where residues which are involved in site 1 interactions can also interact at site 2. And finally, that our hybrid NMR-MD approach, it provides crucial insights into the receptor binding mechanism. And with that, I want to acknowledge our group, which I'm a part of, and also Krishna Rajaratnam's group at UTMB Galveston, where Mohan did a lot of the heavy pulling for the NMR data. And with that, I'll take questions. Thank you.
Thank you. So please type in your questions. Umbrella, any questions? So those two binding sites, are they different in terms of biological functions, or they just have two choices?
No, so the site 1, site 2 is definitely known to be what activates the receptor. Binding at site 2 activates the receptor, whereas site 1, the function was not well established, but we seem to see that it might be involved in the specificity of the ligands.
Okay. Thank you. No more questions, and it's a perfect timing. That's the end of this session. So I thank all the speakers and all the attendees, and we'll conclude this session. And Don, do you have anything to say?
Yes, thank you very much. And I want to remind people that there'll be a poster session now, and we have 40 minutes for posters, so it's not a long time, but we think it'll be valuable to you to join the posters. And before Don gives us a demo of how to do that, I wanted to remind you, tomorrow we have our computational modeling session. And, you know, like today, where at least I learned many new things about single molecule experimental methods, I think computational modeling has a number of stunning advances that go from the systems level all the way to protein design and prediction of fold. And we're at the pivot point where these computational advances will immediately feed into experimental advances like the cryo-EM that we'll cover Friday. So Dawn, do you want to give us an intro to the posters, please?
Sure. Okay. Posters, super easy. They're on the left side of your screen. You can click on any of the titles you're interested in. You can see the poster, you can make it larger or smaller. You'll find out about the presenter. This has the abstract. Feel free to chat here. And the poster presenters each have their own individual Zoom room. They'll be in there from now until 12:40, both today and tomorrow, with their presentations. So please take time, go visit all of our poster presenters. We have some great posters for this conference.
Great, thanks. Join the posters, and we look forward to seeing you tomorrow and to having more discussion. Thanks very much.
Thank you.